If you can spare a hand for a moment, left or right, it doesn't matter. Hold it up and turn the inside of the wrist toward your face. Now bring the tip of the thumb to the tip of the pinky and bend the wrist forward, just a little, like you're trying to make the two fingers meet over the inside of the forearm. Look at the underside of the wrist, if you can see it. If you can't, if the eyes are already closed, imagine the inside of your own wrist from memory. In most of us, a thin cord rises up under the skin when the hand is shaped this way, running a few centimeters down the center of the forearm. It shows only in this position. Most of the time, it isn't visible. Most of the time, you never think about it. You may not see anything at all. In roughly 1 in 5 people, there is nothing there. The cord is gone. The proportion varies with population and ancestry, sometimes wildly. In East Asian populations, it's closer to 1 in 20. In parts of the Arab Middle East, it can be closer to 1 in 2. The wrist is smooth either way. Whatever you are looking at or not looking at is where tonight begins. The cord, when present, is called palmaris longus. It is a slender muscle that runs from the inside of the elbow down to the heel of the hand, where its long, thin tendon fans out and fades into the sheet of tissue that holds the palm together. It does almost no measurable work. Surgeons remove it when they need a tendon to repair something more important elsewhere, a torn flexor in the hand, a damaged ligament in the knee. The people they take it from don't notice it is gone. Their grip is the same. In the primates we descended from, palmaris longus mattered. Those ancestors held their weight on the inside of their wrists, swinging from branches, grooming, climbing, working food out of bark with hands that had not yet learned to stay on the ground. The muscle had work to do. It has been several million years since it did that work. Our lineage left the trees. The selection that maintained the muscle relaxed, and the muscle has been quietly thinning out of the human population, unevenly at different rates in different places, ever since. What you are looking at when you can see the cord is the last light on a muscle that is on its way out. This is the first entry in a record that is inside you. Every muscle, every nerve, every fold of skin, every stretch of DNA carries the fingerprint of something that was selected for, or against, or neither, in an ancestor you will never meet. Some of what you carry still works. Some of it has been reused for a task that has nothing to do with the pressure that first built it. Some of it does nothing and stays where it is because removing it would cost more than leaving it. And some of it is a ruin, a gene still sitting in every cell of your body, broken 60 million years ago in a single ancestor, silent ever since. The first instinct, when people hear this, is to think of these things as flaws. That framing is very old, and it is very wrong. The body is no more unfinished than geology is unfinished, or language is unfinished, or the record of a long night's rain is unfinished. It is a record, and that the record exists in you at all is not a statement about your importance. It exists because nothing erased it. Before we go further, one small thing. If what you are listening to tonight feels worth being made, the subscribe button is the biggest help a channel like this one can receive. And in the comments, I'd like to know something. What is the one thing about your own body that has felt, for a long time, like it shouldn't be there? I read the answers. I find that question more interesting than almost any data in this field. Now, if you are comfortable, get a little more comfortable. Your jaw is probably tighter than it needs to be. Your breath will lengthen on its own over the next few minutes. You don't have to help it. In a moment, your skin is going to do something small and involuntary. A shift in temperature, a line in the music, a memory arriving without warning. And what happens in your body when it does has been happening in mammals, in just this way, for roughly 200 million years. It has happened at least once already today, almost certainly. The air shifted in a cool room, and before you consciously noticed the drop in temperature, the skin along your forearm had already reacted. A faint tightening, the hair rising in ragged little waves across the surface, a few seconds of a texture you could not produce on purpose if you tried. Or it arrived with music, some small harmonic turn in a song you thought you knew, and the back of the neck tightened, and the hair along the top of the spine lifted for a beat, and you felt the listening body itself change. Or it was a memory, a particular person, or a phrase someone once said to you, arriving without permission, and the skin responded before the mind had finished the thought. Whatever set it off, the mechanism is the same. Beneath the surface of the skin, at the base of nearly every hair on your body, there is a tiny strip of smooth muscle called the arrector pili. It runs at a slant from the side of the follicle up to just beneath the skin. When the sympathetic nervous system releases a pulse of noradrenaline, which is what it does reliably and rapidly in response to cold or a startle or a shift in emotional state, the muscle contracts, the follicle tilts upright, and the hair stands up. In almost any other mammal, what this is doing would be obvious at a glance. A cat's fur rising along the spine is one of the most legible communications in the natural world. Double the apparent size, warn the approaching threat. A dog shivering in a cold yard does the same thing more quietly, the raised fur trapping a thin layer of warmer air against the skin. In a squirrel, in a bear, in a fox, in almost any small furred body caught in a cold dawn, the rising of the coat is a working response to a working problem. On you, the same muscle fires the same way. The nerves are the same. The neurotransmitter is the same. The timing is identical. You can feel it happening right now if your attention catches it. What you cannot feel is the fur it is trying to lift. Your lineage lost most of its body hair somewhere between 1 and 2 million years ago, probably on the savannas of East Africa, under a sun that rewarded a body that could sweat efficiently over a body that could insulate. Exactly what drove the loss is still debated. One line of argument favors endurance running and the need to shed heat during long pursuits of prey. Another emphasizes the weight of ectoparasites that a thick coat would have carried. A third argues that sexual selection did much of the quiet work. The honest answer is that more than one of these forces was probably involved, and the sequence in which they acted has not been fully reconstructed. What is clear is the outcome. Our ancestors stopped being a furred animal. Our skin became the exposed surface it is today, and the system responsible for lifting the fur did not go anywhere. Selection cannot remove a muscle simply because the pressure that once shaped it has eased. Selection can only remove a muscle if keeping it costs the organism more than losing it would. Arrector pili are cheap, and they are harmless, and so they stayed. By the end of the 20th century, that was the entry you would have found in any anatomy textbook, in almost those words: a vestigial smooth muscle associated with the hair follicle, responsible for piloerection in furred mammals, retained without identified current function in humans. It was a leftover, small and dutiful, and quietly pointless. And for most of a century, no one had a reason to look at it again. In the summer of 2020, someone did. A group working under Ya-Chie Su at Harvard, in collaboration with the laboratory of Sungjian Lin at National Taiwan University, published a paper in the journal Cell with the kind of title that tends to understate its own findings. What they had done, using electron microscopy at a resolution fine enough to catch individual synaptic structures, was photograph the point of contact between the sympathetic nerve and the base of a mouse hair follicle. And they had found something no one had quite expected to see there. The nerve was not simply passing by, it was forming synapse-like structures directly against the stem cells of the follicle, the cells responsible for generating new hair. The architecture of it mattered. The erector pili muscle, it turned out, was acting as a structural bridge, holding the sympathetic nerve fiber in position against the follicle so that the stem cells sat within reach of the neurotransmitter the nerve releases. Without the muscle to anchor the nerve drifted, and the stem cells fell quiet. With the muscle in place, the whole system held together, and the stem cells remained responsive. When the cold came then, and the nerve fired, the noradrenaline it released was doing two things at once. It was contracting the muscle, which produced the goosebumps, the old mammalian response to a lost coat, and it was washing across the stem cells of the follicle, which responded by waking out of quiescence and beginning to divide. In mice held at cold temperatures for 2 weeks, the scientists watched new hair grow. This is not a complete rewrite of the textbook entry. The piloerection function itself, the thermoregulatory raising of fur in response to cold, is still gone in humans. Our fur is too sparse for the rising of individual hairs to make any meaningful difference to body temperature. You can raise every hair on your body and your skin will not be warmer for it. That part of the story is intact. The muscle you felt respond a few moments ago was firing to lift a coat that is not there, and that will not grow back, and that has not been there in any of your ancestors for longer than our species has been a species. What has changed is the surrounding picture. The system the original selection abandoned has not been idle. The nerve that innervates the muscle is also, it turns out, the nerve that tells the hair follicle it is time to regenerate. The muscle holds the nerve in position. The muscle was retained because it cost the body very little and because the body happened to have further uses for the scaffolding it provided. The function for which selection originally shaped the erector pili is, in you, a vestige. The function it has come to serve in parallel is not. Selection did not plan this. Selection does not plan anything. The second function came into view only because the structures it needed happened to already exist, assembled under a different pressure entirely, and because a nerve passing through a tissue is, from the nervous system's point of view, an opportunity. The muscle became useful for a second task, not because any selection pressure demanded the task be solved, but because the components were already there and nothing had ever unbuilt them. There is a version of this story we do not usually tell ourselves about the body. We tend to imagine, when we imagine evolution at all, that each part either earns its place by serving a current function or is slowly erased by selection once that function ends. The erector pili complicate both halves of the picture. The original function ended. The structure was not erased, and while the structure stayed on, apparently doing nothing, the organism quietly turned it to another use that no selective pressure had specifically demanded. The goosebumps rising on your arm tonight are an old program running on old hardware. The hardware, it turns out, is running something else at the same time, and the arm carries more than one program. Somewhere between the inside of the elbow and the heel of the hand, threaded through the body you are listening with, there is a muscle that belongs to a different chapter of your lineage entirely, not the mammalian one that gave you the fur, but the primate one that took your ancestors into the trees. In some people the muscle is present, in others it is missing entirely. Whether you have it at all is a question you can answer tonight without getting up. Textbooks tend to describe palmaris longus as a slender forearm flexor of variable presence, useful to surgeons as a tendon graft, and then move on. What the standard description underplays is how variable the muscle actually is. Not variable in the small way that most anatomical features are variable, with minor fluctuations around a general pattern, but variable in a way that no other muscle in the human body comes close to. Taken across 7 decades of studies, in populations on every continent, with tens of thousands of wrists examined by dissection, ultrasound, or physical test, The proportion of people missing palmaris longus entirely ranges from around 1.5% at the low end to nearly 64% at the high end. A gap that wide in a structure that every anatomy textbook still describes as a single standard part of the forearm is the sort of anomaly that usually means something about the structure itself is on the move. The variation is not random. It tracks population and ancestry, and when the figures are laid out on a map, they look a great deal like something else. The lowest rates of absence, the populations, that is, in which the muscle is still most reliably present, appear in East and Southeast Africa, where palmaris is gone in only about 1 in 40 people. In populations across the rest of the continent, the rate climbs to roughly 1 in 9. In East Asia, a different pattern altogether, with the muscle gone in about 1 in 20, which is itself a long way from the European figure of more than 1 in 4. In Turkish populations, close to 1 in 3. In parts of the Arab Middle East, the muscle is gone more often than it is present. A biologist named Kaysar Yamine, working out of the Lebanese-American University's Medical School in Beirut, put together the most rigorous meta-analysis of this distribution in the early part of the last decade. What Yamin noticed, when the figures were set next to each other, was that the population with the lowest rate of absence, the population in which the old selection still appeared to be holding the muscle in place, sat at the geographic origin of the species. The populations that had moved out of Africa over the last 60 or 70,000 years, that had spread across the continents and adapted to every climate and ecology they encountered, were the populations in which the muscle was going away fastest and most variably. The pattern does not, on its own, prove a causal story. What it shows is a signature. In the lineage that stayed closest to where the species began, something about the conditions of life appears to have been holding palmaris more firmly. In the lineages that spread, that hold loosened, and the muscle has been drifting out of the species along the way. What palmaris had been selected for, deep in our primate ancestry, was holding on. In the monkeys and apes we descended from, the inside of the wrist carried a significant share of body weight during arboreal movement, swinging between branches, reaching across gaps that could not be walked, pulling the body upward through a vertical world. Palmaris was one of the muscles contributing to the grip that let an ancestor hold a limb long enough to swing from it. It was not the only one, but it was one of them. Somewhere between 6 and 8 million years ago, the lineage that became us left the trees. The ground became the place the hominid body carried its weight, and the forelimbs stopped being organs of suspension. They became something else: tool users, carriers of infants, throwers of stones, shapers of language through gesture. The specific contribution palmaris had been making to a grip that held a body against gravity stopped mattering. What happens to a muscle whose contribution is no longer enforced by selection is not that it is actively removed. Selection has no forward-looking editorial function. Selection cannot identify a structure that has outlived its usefulness and send an instruction for its erasure. What selection can do is stop actively maintaining the structure. When the maintenance pressure lifts, the developmental program that builds the muscle is free to accumulate mutations that would previously have been trimmed back. Some of those mutations, when they occur, produce an individual in whom the muscle simply does not form. In a population where the muscle was still doing work, such an individual would have been at some small disadvantage, and the mutation would have been filtered out of the gene pool within a few generations. In a population where the muscle was not doing work that mattered, the individual is indistinguishable from their neighbors and the mutation stays. Over thousands of generations, the proportion of people missing the muscle drifts upward, and the drift happens at a rate that depends on local history, on the size of the population, on the pattern of its mating, on the bottlenecks it has passed through, on sheer statistical luck. This is the process you are looking at or not looking at when you check the inside of your wrist. The cord you saw earlier, or the absence of it, is a single reading from a slow statistical movement that has been underway for several million years and is still underway now. If your wrists are smooth and the cord did not rise, you belong to a lineage in which the muscle has been drifting toward absence a little faster than in some others. If the cord rose, you belong to a lineage in which it has been drifting more slowly or not at all. Neither condition tells you anything about your strength or your ancestry in a direct sense. What it tells you is that you are one point in a distribution, and the distribution is moving. In the meantime, the muscle has a second life that has nothing to do with the tree it no longer grips. In almost any hospital with a hand clinic, the scenario plays out several times a week. A patient arrives with a flexor tendon severed in the palm. A kitchen accident, a fall onto broken glass, an industrial injury that tore through the long flexor of a finger. The hand is swollen. The finger cannot curl. The surgeon scheduled for the repair needs a length of tendon to replace what has been lost, and before the patient is brought into the operating room, a junior doctor performs the same small gesture you did earlier. Thumb to pinky, wrist flexed, a look at the forearm. If the cord rises, the decision is made. The procedure will begin at the inside of the wrist. A short incision opens the skin. The pale, thin tendon of palmaris is lifted clear of the larger flexor next to it. The muscle is detached from its origin at the elbow, and the freed tendon becomes raw material. It is routed forward into the hand, where the ends of the lost flexor are waiting, and it is stitched in as a biological bridge. A few centimeters of reclaimed tissue doing the work the original had been doing for decades. The patient keeps the grip they arrived with. The muscle is gone entirely. They never notice. The structure a primate ancestor needed to hold a branch becomes, in the right operating room, the thing that lets a carpenter hold a hammer again. There is no designer in this story. A designer would have either preserved the muscle at its original task or arranged for its removal to be complete and uniform. What the body shows instead is a muscle that is neither a structure still forming in most people, still named, still teachable, still present in the anatomy textbooks, still being harvested every week to repair injuries it had no selective relationship to, and still quietly going away in different places at different rates, generation by generation. It is small, and it is slow, and if you did not know it was there, you could live your whole life without thinking about it. What comes next is not small. A single nerve, thicker than the tendon you may or may not have at your wrist, descends from the base of your brain into the top of your chest, wraps around the biggest artery in your body, and climbs back up through your neck to reach the voice box you are using when you form the words of this sentence in your mind. In a human being, the journey takes about half a meter. In a giraffe, it is close to 5. And the reason it does this, instead of taking the direct path that would be 5 centimeters long, is the kind of history the body cannot hide. In the spring of 2009, a film crew from a British documentary program called Inside Nature's Giants set up in an anatomy theatre at the Royal Veterinary College on the edge of London. The subject on the table in front of them was a juvenile female giraffe, about 4.5 meters tall, that had died a few days earlier at a safari park in Bedfordshire. The necropsy was being conducted by Joy Reidenberg, a comparative anatomist from the Icahn School of Medicine at Mount Sinai in New York, and she had invited Richard Dawkins down for the afternoon to watch a specific moment in the dissection. The moment concerned a nerve. By the time Dawkins arrived, Reidenberg had opened the animal's neck from the base of the skull down toward the chest, and she was working her way along one particular bundle of fibers on the left side. The nerve they were tracing is called the recurrent laryngeal, and it begins where you would expect a nerve that controls the voice to begin, at the base of the brain, branching off one of the cranial nerves called the vagus. From there, though, it does something strange. instead of dropping the short distance from the base of the brain to the larynx, which would be a matter of a few centimeters in the giraffe and perhaps half that in a human, the nerve dives downward through the neck in the opposite direction, traveling alongside the trachea all the way to the thoracic cavity. In the chest, at the level of the heart, it loops around the arch of the aorta, the largest artery in the body, and then begins the long climb back up the neck it has just descended, eventually reaching the larynx from below. In the giraffe on the table that afternoon, Reidenberg and her students pulled the nerve free of the surrounding tissue section by section, laid it out alongside the animal as they went, and measured it. From the base of the brain down to the aortic arch, back up to the voice box, the nerve ran for very close to 4.5 meters. Fully half of that length was devoted to reaching a structure that sat, in a straight line from its origin, no more than a handful of centimeters away. What Reidenberg was showing Dawkins, and what the cameras were there to record, was a structure that looks, when you see it laid out in full, like a mistake. It is the kind of structure that a human engineer, given the job of wiring a brain to a voice box, would not have produced. No one designing the system from scratch would choose to route a control cable halfway down the body and back up again when the target sits a finger's breadth from the source. The route is not short because it could not be short. The route is long because of something that happened to an ancestor. That ancestor was a fish. The nerve that becomes the recurrent laryngeal in modern tetrapods is the same nerve that, in the early jawed vertebrates of about 400 million years ago, innervated a section of the gill arches. In a fish, the layout makes perfect sense. The nerve leaves the brain, runs behind the developing arterial arches that supply the gills, and terminates in the tissues it serves. There is no loop because there is no need for one. The nerve passes behind a vessel on its way to its target, and its target is right there. What happened over the long stretch of time between that fish and the giraffe on the dissection table is that the vertebrate body plan began to pull itself apart in the front. The gills disappeared. The arterial arches that had supplied them were repurposed. One pair became part of the systemic circulation, another contributed to the pulmonary circuit, and the one the nerve had originally passed behind became, in mammals, the aortic arch. The heart migrated downward, away from the head into the chest. The neck lengthened. And the nerve, which in the embryo is laid down before any of this rearrangement takes place, stayed behind the structure it had always been behind, which meant that as the aortic arch descended into the thorax, the nerve descended with it. The original short path was preserved as a developmental sequence. It just happened, in an animal with a neck, to turn into a detour. There is no mechanism by which the nerve could have been rerouted once the body plan changed. By the time an embryo is old enough for its neck to begin lengthening, the recurrent laryngeal is already in position, already looped around the aortic arch, already committed. To reroute the nerve would require a wholesale revision of the order in which the embryonic tissues are laid down, and revisions of that magnitude are not available to selection. What selection can work with at each generation is only small variations on the developmental program that has already been running successfully for millions of generations. Disrupting the sequence would kill the embryo. Accepting the detour costs the organism a few grams of extra nerve fiber and a slightly longer reaction time in the vocal cords. The detour is chosen every generation because the alternative is not being born. In a human being, the total length the signal has to travel from the brainstem down into the upper chest, around the aortic arch and back up the neck to the larynx works out to something close to 0.5 meter. In a giraffe, as Reidenberg demonstrated that afternoon, it comes out to nearly 5. The difference is not a difference in architecture. The architecture is identical. It is a difference in how much neck the architecture has to cross, and giraffes have a great deal of neck. There is a logical extension of this which the paleontologist Matthew Weddell worked through in a paper published in Acta Paleontologica Polonica in 2012. If the recurrent laryngeal takes the same route in every tetrapod, and if that route scales with the length of the neck the animal carries, then the longest-necked animals that ever lived should have had the longest recurrent laryngeals that ever existed. The longest-necked animals that ever lived were the sauropod dinosaurs, the brachiosaurs and diplodocoids and titanosaurs of the Jurassic and Cretaceous, some of which carried necks of 10 meters or more. Weddell ran the calculation for Supersaurus, one of the largest sauropods in the fossil record, and arrived at a figure of approximately 28 meters for the total path length of its recurrent laryngeal nerve. A single nerve cell in a single animal nearly 100 feet long. Among the longest cells by any plausible definition in the history of life on Earth. That cell existed because a particular Jurassic dinosaur inherited from a Triassic ancestor, which had inherited from a Carboniferous ancestor, which had inherited from a Devonian fish, a developmental sequence in which a nerve is laid down behind an arterial arch before the neck has begun to grow. The fish had not needed a long nerve. The dinosaur grew one anyway, and every tetrapod alive today carries the same arrangement, scaled to its own particular dimensions. You are carrying it right now. If the ambient temperature of the room were to drop suddenly and you wanted to say so aloud, the instruction to shape the words would leave the motor cortex, descend through the brainstem, enter the vagus nerve, pass into the chest, loop around the top of your aorta just above the heart, and climb back up the neck through the muscles of the throat to reach the larynx, where the vocal folds would respond. The signal travels approximately 50 centimeters to reach a voice box that sits in a straight line from its origin, close enough to touch. The speed is fast enough that the delay does not matter for speech, but the path is longer than any engineer, given the problem, would have allowed. Because the path was not allowed, the path was inherited. There is a clean way to state what the recurrent laryngeal nerve actually is, and it is the statement that stays with people who see the giraffe dissection for the first time. The recurrent laryngeal nerve is not a design, it is a history. It records in the geometry of a single cell the order in which a fish became a tetrapod became a mammal grew a neck, and a neck stretched until the original root had become the scenic root. You cannot remove the history by any means short of starting the organism over from a single cell, and the single cell, when you start it, begins by laying down the nerve behind the arterial arch again, exactly as the ancestor did. This is, according to some estimates, the clearest piece of direct anatomical evidence for the way evolution actually works, for the fact that natural selection does not redesign, that it only modifies what was already there, that the body is constrained at every turn by the history of the bodies that came before it. Daniel Dennett has called it the best single argument against intelligent design he has ever encountered. Dawkins, in his own account of watching Reidenberg trace the nerve through the giraffe that afternoon, described it as the kind of evidence that does not need a frame around it. You see the nerve. You follow it. You understand what it is telling you. What it is telling you is inside your chest right now, and it will be there every time you speak tonight. It will also, if you are quiet enough in a quiet enough room, be telling you something about a much older structure. Something older, in fact, than any nerve or muscle in your body. A structure that most biology textbooks, for most of the last century, described in a single dismissive phrase as an evolutionary vestige, useless, sometimes dangerous, routinely removed, and almost certainly on its way out of the species. Darwin thought that about it. Most doctors still do. The evidence has quietly turned against both of them. There is a short passage in the 3rd chapter of The Descent of Man, published in 1871, in which Charles Darwin offers the appendix as an example of what he calls a rudimentary organ. He does not spend very long on it. He notes that the human appendix, that small blind-ended pouch that hangs off the beginning of the large intestine, appears to be a shrunken remnant of a much larger structure found in other mammals. A fermentation chamber, he thought, used by herbivorous ancestors to break down tough plant material and inherited by us in a reduced and functionless form. He adds, almost in passing, that the organ is prone to occasional fatal inflammation, which he treats as evidence of its useless and even dangerous character. Then he moves on. This passage has done an enormous amount of work over the last century and a half. It appears in biology textbooks, in popular science books, in museum exhibits, in classroom lectures. It is the reason almost everyone who has been to a high school in an English-speaking country can tell you, if asked, that the appendix is a vestigial organ, a leftover, a useless remnant, an evolutionary curiosity that occasionally needs to be cut out before it kills you. Darwin said so, the textbooks say so, and for the whole of the 20th century very few people had reason to look at the claim any more closely than that. The claim, it now appears, is wrong. It is not wrong in the way that a completely discredited idea is wrong. Darwin was right that the appendix is smaller in humans than in many other mammals, right that its precise function was mysterious, right that inflammation of it could be fatal. What he got wrong was the conclusion. The appendix is not a shrinking remnant on its way out of our lineage. It is a retained structure that has been quietly selected for across the mammalian family tree for at least the last 80 million years. The reconsideration began in 2007 in a surgical laboratory at Duke University in North Carolina. Two researchers working there, a surgeon named Randall Bollinger and an immunologist named William Parker, had been looking at biofilms along the human gut, the dense communities of commensal bacteria that coat the lining of the intestine and play an essential role in digestion and immune function. What they noticed when they examined the tissue of the appendix specifically was that the concentration of this beneficial biofilm was unusually high there. The appendix was, in the language of their 2007 paper in the Journal of Theoretical Biology, a kind of refuge, a narrow, mostly sealed-off pouch in which a population of useful bacteria could maintain itself, safely out of the way of the main flow of the gut contents. The clinical significance of this was not obvious at first, but it became clearer with a few years of further work. During a severe case of diarrheal illness, the contents of the large intestine are flushed out, and the commensal bacteria that live there are flushed out along with them. The colon afterwards has to be recolonized. In most healthy individuals, this happens quickly, with the surviving bacterial population dividing and spreading back out over the gut lining within days or weeks. But if the starting population is too small, or if the diarrheal episode was severe enough to leave the gut almost sterile, the recovery can go badly. The colon can be recolonized instead by opportunistic species that take advantage of the empty territory, and the result is a chronic and sometimes life-threatening disruption of the gut ecosystem. In people who have had their appendix removed, Parker and his colleagues found this scenario is measurably more likely. The risk of recurrent infection with Clostridium difficile, an opportunistic pathogen that causes exactly this kind of disrupted gut syndrome, is roughly twice as high in individuals without an appendix as in those who still have one. Subsequent studies have turned up similar associations with several other conditions involving the gut ecosystem, including some forms of inflammatory bowel disease and, more recently and more speculatively, with conditions like Parkinson's that are now thought to begin in the gut. The associations are subtle. They are not catastrophic. An appendectomy is not, as some early reports implied, a kind of hidden harm done to the patient. but the idea that the appendix is a purely useless organ whose removal has no consequences at all is not supported by the data. Something is being lost in small statistical amounts when the appendix is taken out. This alone would have been enough to reopen the textbook entry. What made the reopening unavoidable was a second line of work run in parallel on the evolutionary history of the structure. Heather Smith, an anatomist at Midwestern University in Arizona, assembled with Parker and a French paleontologist named Michel Laurin the largest comparative survey of the appendix that had ever been attempted. They looked at 533 species of mammals, traced the presence or absence of the appendix across the phylogenetic tree, and reconstructed the most likely history of the organ across the mammalian family. The result, published in a series of papers between 2009 and 2017, was a surprise. The appendix, on their analysis, has evolved independently at least 32 separate times within mammals. It appears in Australian marsupials, in rodents, in rabbits, in several lineages of primates, in tree shrews, in a handful of carnivores. It does not appear in a single common ancestor and then get inherited or lost at random across the descendants. It appears over and over in lineages that diverged hundreds of millions of years before the appendix they each carry came into being. The structure is not a single evolutionary event. It is a solution independently arrived at by many separate lineages of mammals under selection pressures that evidently favor its emergence, and once it emerges, it tends to be kept. The total age of the appendix across the mammalian tree works out to at least 80 million years of accumulated evolutionary presence. That is not the signature of a vestige. That is the signature of a structure doing something useful enough that selection maintains it, and useful enough that it has been reinvented in slightly different forms in dozens of mammalian lineages that never shared it. The common theme, when Smith and her colleagues looked at what united the species that carried an appendix, was the one Bollinger and Parker had identified from the clinical side. The appendix-bearing mammals consistently had a greater concentration of lymphoid tissue, the specialized immune tissue that produces antibodies and supports the gut's interface with its bacterial residents, than their appendix-less relatives. Whatever the precise function of the structure turns out to be in any given lineage, it involves immunity and it involves bacteria. Darwin's proposed function, a shrunken fermentation chamber left over from a plant-eating past, is not the function selection has been acting on. Selection has been acting on something else entirely. What makes this case particularly worth sitting with for a documentary about the body's evolutionary leftovers is that it is a reversal of a reversal. The first time the appendix entered the public imagination as an evolutionary story, it did so as the classic example of vestigiality, the clean textbook case, the one Darwin himself signed off on. It was the structure everyone pointed to when they wanted to show that the body carries useless remnants. Over the course of the last 2 decades, the evidence has quietly turned around, and the classic example has become something different. An example of how a function that goes unrecognized for a long time can still be holding an organ in place, generation after generation, without anyone realizing that the selection pressure is still active. The appendix is not a vestige that evolution forgot to remove. It is a solution that evolution keeps rediscovering. The implication for the frame this documentary has been building is worth naming directly. Not every small, obscure, occasionally troublesome structure in the body is a leftover. Some of them are. Some of them, as we will see, are genuinely ruined. Structures whose original function is gone and whose present function is either absent or minor. But some of them, like the appendix, are working organs whose function was simply not visible to the observers who named them useless. The body archive is not a single kind of record. It is a layered document, and some of what looks like vestigial scrawl in the margin turns out, on closer reading, to be doing quiet load-bearing work. You are carrying the vermiform appendix, most likely, unless a surgeon has removed it at some point in your life. If you still have it, it is hosting, at this moment, a biofilm of commensal bacteria that are indistinguishable from the ones in your colon, except that they are better protected. If it has been removed, your gut ecosystem is functioning without that reserve, and you are slightly more vulnerable to the kind of disruption that the reserve exists to buffer against. Neither condition is dramatic. Both are visible when you know how to look. The appendix was not the only structure the 20th century labeled a useless leftover on the basis of limited evidence. Another structure carries a more complicated kind of label. Not quite vestigial, not quite functional, but caught somewhere in between, and the subject of an active scientific argument about what, if anything, the right name for it is. It lives further down the body, near the base of the calf, and roughly 9 people out of 10 still have it. The largest tendon in the human body lives at the back of the heel. It runs from the combined muscles of the calf down to the bone. The ankle pivots on, thick enough to see under the skin when the foot is flexed, strong enough to bear several times the body's weight when the walk quickens into a run. It is called the Achilles. Almost every adult can name it. Running alongside the Achilles, threading down the back of the calf just medial to it, and inserting into the heel only a finger breadth away from where the larger tendon inserts, is a much thinner cord belonging to a much smaller muscle. In most of us, it is not palpable from outside. The calf muscles surround it, the Achilles sits in front of it, and the ordinary experience of running a hand along the back of the leg would not reveal it. The muscle is called plantaris. Approximately 9 out of every 10 human beings have it, which means that, unless the odds have broken the other way, you are almost certainly carrying one on each leg right now. In the 10th person, nothing about the calf's behavior changes. The foot lifts with the same force. The walk is indistinguishable from the walk of someone who still carries it. For the longer part of a century, that was taken as sufficient evidence that the muscle was not doing anything worth the body's trouble. By the standard textbook account, plantaris is a vestige. In the monkeys and apes we descended from, the muscle did real work. Its tendon ran not just to the heel, but all the way to the sole of the foot, where it contributed to grip. Those ancestors used their feet the way we still use our hands. They grasped branches with them. They worked food out of bark, handled objects, held position on narrow limbs with the whole lower body engaged in prehension. The foot below the knee was an organ of grip as much as it was an organ of locomotion. When our lineage committed to walking on the ground, the grasping foot became a walking foot, the sole became a weight-bearing surface, and the muscles that had contributed to grip stopped having grip to contribute to. Plantaris, on this account, is what was left behind. A muscle that over the last several million years lost its reach into the sole, retracted its tendon up to the heel, shrank to near irrelevance, and is now on its slow way out of the species. The trouble with this account is that it has been true for some decades now, and the muscle is not going anywhere. The prevalence figures have been remarkably stable across studies going back more than 50 years. Plantaris appears in about 90 to 93% of the population, almost regardless of which population is examined. It is present consistently in fetal development. The tendon forms on schedule, the muscle belly attaches where the anatomy atlases say it should, and the variants people display in adulthood appear to be variants in the underlying developmental program rather than evidence of progressive loss. If plantaris were vanishing the way palmaris longus has been vanishing, with widening regional gaps, with populations drifting toward absence at different rates, with the signature of a structure no longer actively maintained, that signature would be visible in the data. The signature is not there. Something else has been visible once people began looking for it. The muscle belly of Plantaris, examined under a microscope, turns out to be unusually rich in a particular kind of sensory organ, a tightly wound bundle of nerve and muscle fibers called a muscle spindle. Muscle spindles are the body's internal stretch sensors. They report, continuously and automatically, how much a particular muscle is being stretched at any given moment, and that information travels back to the spinal cord and the brain, where it helps coordinate balance, posture, and movement. Every skeletal muscle in the body contains some number of these spindles. What varies between muscles is the density, the ratio of spindles to contractile fibers. And in most muscles, the ratio is such that the structure is primarily a worker and secondarily a sensor. In Plantaris, the ratio runs the other way. The density of muscle spindles is high enough, relative to the muscle's modest contractile capacity, that the structure looks less like a muscle that happens to contain sensors and more like a sensor that happens to be housed in the form of a muscle. A review published in the journal Biology in the middle of 2025, led by a research team interested in revisiting the Vestigial label, argued that this flips the interpretation of the whole muscle. Plantaris, on the spindle-dense reading, is not a grasping muscle in slow decay. It is a grasping muscle whose role in the body has shifted from a structure that did mechanical work to a structure that does sensory work. The specific sensory work involves reporting, in real time, how the entire posterior compartment of the calf is being loaded during walking, running, climbing, and standing. When the weight shifts from one foot to the other, when a stumble has to be caught, when the balance has to adjust on an uneven surface, the feedback the nervous system uses to orchestrate the correction runs in part through the Plantaris. The muscle that once gripped a branch did not disappear when grip stopped being selected for. Its spindles kept reporting stretch, as spindles do. The difference is where the information goes. The original function relaxed. The hardware kept working. The information the hardware had always been generating found itself funneling into a different problem, one involving a calf under the demands of upright walking rather than a foot in a tree. The case is not closed. A 2025 review is not a final verdict, and the spindle density data do not overturn the older vestigial interpretation so much as sit alongside it awkwardly. What the review has done is make the category of vestigial harder to apply to this particular muscle without additional caveats. Plantaris is not actively being lost from the species. Its original function is no longer performed. It appears to be doing something the original function did not involve. And yet its contribution to modern gait is modest enough that removing it, as surgeons routinely do in Achilles tendon repairs, leaves the patient with a calf that functions indistinguishably from before. The muscle sits uncomfortably between the two textbook categories it is supposed to fall into. The clinicians who harvest the Plantaris tendon have known about its expendability for a long time. In hand surgery, as with palmaris longus at the wrist, it is available as a tendon graft when something more important has been torn. More often, though, it is used in its own native territory, during the repair of a ruptured Achilles, where the Plantaris tendon can be split off from its insertion, routed along the length of the damaged Achilles, and stitched in as a reinforcing strip. If you ever have an Achilles tear repaired surgically, your Plantaris is one of the structures the surgeon will almost certainly consider harvesting along the way. The patient wakes up without a muscle most of them never knew they had. The ankle, once healed, moves normally. Whatever proprioceptive work the muscle had been doing, the nervous system appears to redistribute among the other calf structures without significant loss. What label ought to attach to a structure like this is an open question, and the honest answer is that the category is not quite the right shape for what the evidence shows. Calling Plantaris vestigial implies a decay that is not visible in the data. Calling it functional implies a current role that is real but modest and potentially redundant. The body carries more structures in this ambiguous middle than the older textbook categories could accommodate. You are carrying at least a few of them right now, almost certainly. Plantaris, if you have it, is one. Some of what selection leaves behind is on its way out. Some of it has been put to use in ways the original function never involved. And some of it is simply still there, doing small things, while the surrounding biology settles into what it will become next. Other parts of the body are moving on a very different evolutionary schedule. In the mouth, across a time frame a fraction as long as the one the Plantaris has been drifting through, a frame that covers only the last 2 million years or so, a specific mismatch has been building between two separate developmental processes that had once kept pace with each other and no longer do. It is visible in the present day in the mouths of roughly 4 out of every 5 people alive. In the archaeological record of early Homo, going back roughly 2 million years, a particular category of object begins appearing in large numbers. The objects are small, roughly the width of a modern coin, but a few times thicker, and they are made from stone, most often from volcanic rock or from quartz, depending on what was locally available. Each one carries a working edge that is sharp enough to slice through hide and muscle with some determination behind it, and the edge is produced by striking one piece of rock against another at a specific angle. The technique sounds simple when described, and it requires considerable practice to execute. Archaeologists call the products Oldowan flakes, after the Olduvai Gorge in Tanzania, where they were first systematically described, and they appear in enormous numbers at sites across East Africa for the better part of 1 million years. The hand that made them belonged to a hominin, roughly our height, with a brain about half the size of ours, and a body that was beginning, during the period these flakes were being produced, to change in ways that run deeper than the stone tools themselves. What the flakes were for, in the simplest account, was food. The hominins who carried them were eating more meat than any of their ancestors had. They were also beginning to use the tools on plant material, pounding tubers, slicing fibrous vegetables, processing food in ways that reduced, before a single bite was taken, how much work the jaws would have to do. The specific change this made to a day in the life of an early Homo is not something that has to be reconstructed from indirect evidence anymore. A team at Harvard University worked out, about a decade ago, exactly what it added up to. The work was done by Catherine Zink and Daniel Lieberman, and it was published in Nature in 2016. The experimental setup was straightforward and a little strange. Human volunteers were brought into a laboratory fitted with instruments that measured the muscular effort of their jaws and the electrical activity in their chewing muscles in real time, and given food to eat. The food was not chosen for its culinary appeal. It was chosen to approximate what an early member of the genus Homo would have been chewing 2 million years ago. It included raw goat meat, selected for its toughness and its resemblance to the sort of wild game available on the African savanna. It included root vegetables, beets, yams, carrots, because underground storage organs have always been part of the hominin food supply. The volunteers chewed each food type in several different forms: unprocessed, the way a chimpanzee might still chew it; sliced, the way a stone flake would have left it; pounded, the way a simple rock-on-rock hammering would have left the vegetable material; and in the case of the goat meat, cooked, though cooking would not become widespread in the human lineage for another 1.5 million years after the Oldowan flakes first appeared. The volunteers chewed, and the instruments recorded. After each mouthful, instead of swallowing, the subjects spat the food into a dish, where the sizes of the resulting particles were measured and catalogued. What the data showed was specific and consistent. With a diet composed of about 1/3 meat, which is a reasonable approximation of what early Homo was eating, and with the meat and vegetables processed before ingestion using the techniques a stone flake makes possible, The total muscular effort of chewing dropped by approximately 26%, and the number of chews required to process a day's food dropped by approximately 17%. Those reductions do not sound dramatic in isolation. Extended across an adult lifetime, they amount to something on the order of 2.5 million chews per year that an early Homo did not have to perform because they had a flake in their hand. What the jaw did not have to do, the jaw eventually stopped being built to do. The fossil record of Homo erectus, the species that inherited the older one tradition and refined it, shows a sharp reduction in the size of the jaws and teeth relative to every earlier hominin. The molars shrank. The chewing muscles shrank with them. The whole architecture of the face, brow, cheekbones, the width of the mandible, the depth of the chin, became lighter and narrower over a span of geological time that was, by the standards of major anatomical change, a short one. The driver, on the Zink and Lieberman account, was not cooking. Cooking came later, perhaps as recently as 500,000 years ago, in anything resembling its modern form. The initial reduction was mechanical. Stone tools did most of the work that larger teeth had been doing. The teeth themselves, with the selective pressure that had maintained their size now eased, began to reduce in size and continued reducing across the whole tenure of Homo erectus on the landscape. What they did not do, at least not at the same pace, was reduce in number. The program that builds a human jaw and the program that builds the teeth that fit into it are encoded separately in the developmental genome. The two programs are coordinated, in the ordinary case, tightly enough that the number of teeth produced corresponds to the amount of jaw available to hold them. When selection acts on jaw size, however, when a selective pressure favors individuals whose jaws are slightly smaller than their parents, the jaw size program responds more readily than the tooth count program. Making a smaller jaw is a matter of scaling down an existing structure, which the developmental system can do incrementally and without much disruption. Changing the number of teeth the jaw is instructed to produce is a deeper edit, involving the specific switches that tell the embryonic tooth germs whether to form at all. That edit can happen, and has happened in many lineages across evolutionary history. It simply tends to happen more slowly than the scaling change it is trying to keep up with. Over the last 2 million years, the human jaw has shrunk faster than the developmental instructions for tooth formation have been revised. The result is visible in almost any dental clinic in the world. Between the ages of 17 and 25, the last 4 molars in the human mouth, the third molars, colloquially the wisdom teeth, attempt to erupt. In approximately 80% of young adults, at least one of these teeth cannot complete the eruption because the jaw that is now supposed to accommodate 32 teeth no longer has the room for them. The teeth become impacted. They press sideways into the adjacent molars, or they never clear the gum line at all, or they emerge partially and create a pocket where food and bacteria collect. The surgical profession that extracts them from otherwise healthy young people is one of the largest elective procedures performed in the developed world. The second visible consequence runs in the other direction. In a significant and growing fraction of the population, one or more of the third molars never forms in the first place. The tooth germ that should have been laid down in the embryo is absent, and when the adult mouth reaches the age at which those teeth would erupt, there is simply nothing there. Global estimates place the prevalence of at least one congenitally absent third molar at roughly 22%. The rate varies enormously between populations, close to 40% among Koreans, approaching complete absence in some indigenous Mexican groups. What the variation suggests is that the tooth count program is finally beginning to close the gap with the jaw size program, and in some lineages is closing it faster than in others. Whether the gap will close completely, and how soon, is a question the statistical process of selection has not yet resolved. None of this is the signature of a badly designed structure. There is no engineer who forgot to reduce the tooth count when the jaw was reduced. What the record shows is two developmental programs that were synchronized for the greater part of mammalian evolutionary history, that were placed under a new selection pressure by a behavioral innovation roughly 2 million years ago, and that have been responding to that pressure at different rates ever since. If the listener has had wisdom teeth extracted, surgery has intervened in a process selection has not yet finished. If the listener never had wisdom teeth in the first place, the developmental revision has, in their lineage, run ahead of the jaw reduction rather than behind it. Either way, the jaw the listener carries is the intermediate form of a transition that has been running for approximately 100,000 generations and will continue at whatever pace selection allows for as long as the lineage continues. The transitions that shape the human body run at many scales. Some of them, like the jaw-tooth mismatch of the last 2 million years, are visible in bone and in the ordinary geometry of a mouth. Others are buried deeper, down in the arrangement of single stretches of DNA, measurable only because the sequencing techniques of the last 20 years have made it possible to read them. A particularly specific transition of this kind, a change recorded not in the shape of a skeleton, but in the arrangement of a few hundred nucleotides on one chromosome, happened roughly 25 million years ago, and the evidence for it is something you are sitting on this moment. If you were able to look inside a human embryo around the 5th week after conception, before any of the structures we would call characteristically human have clearly formed, you would see, among many other things, a tail. It is not a vestige of a tail or a suggestion of a tail or a symbolic gesture toward the idea of one. It is a tail. It extends from the posterior end of what will become the spinal column, and it contains, depending on the particular embryo, something between 10 and 12 small vertebrae laid out in the curve you would expect a tail to take. It has the proportions of a tail. Its blood supply is organized the way a tail's blood supply is organized. It is anatomically indistinguishable in those first weeks from the embryonic tails of every other mammal at the equivalent developmental stage. Over the next several weeks, the tail is taken apart. The process begins with a change in the expression of a suite of genes that had been supporting the tail's growth, and it proceeds through a mechanism called programmed cell death, in which the cells that make up the posterior portion of the tail receive chemical instructions to destroy themselves. The destruction is tidy. The cells dismantle their own internal machinery, are engulfed by surrounding tissue, and are recycled. The blood vessels retract. The nerves that had been extending into the tail withdraw. By the end of the 8th week, most of the tail is gone. What remains is a fused column of 3 to 5 vertebrae at the base of the spine, locked in place by the time the embryo has any recognizable human shape. And those vertebrae will eventually become the structure called the coccyx. It is the floor of the pelvis. It is the anchor point for the muscles that hold the pelvic organs in place. It is the thing you are sitting on right now. The question of why our ancestors lost their tails and when is one that anatomists and evolutionary biologists have been circling for most of the past century. What could be said with certainty until quite recently was that the loss was old. Older than the split between humans and chimpanzees, older than the split between our shared lineage and that of gorillas, older than the emergence of the first upright walking hominins. The tail was already gone by the time the hominoid line, the branch of primates that includes the great apes and the gibbons, emerged into a fossil record distinct from the old world monkeys. A reasonable estimate, based on the molecular and morphological evidence, placed the loss somewhere in the neighborhood of 20 to 25 million years ago. What could not be said until 4 years ago this month was how it happened. In February of 2024, a group led by Bo Xia at the Broad Institute and Itai Yanai at NYU Langone published a paper in the journal Nature that answered the question with a specificity no one in the field had quite expected. What Scheer and his colleagues had done was compare the genomes of apes, which lack tails, against the genomes of old-world monkeys, which still have them, looking for any genetic difference that might correlate with the presence or absence of the structure. The difference turned out to live in a gene called TBXT, which codes for a transcription factor, a protein that regulates the activity of other genes, essential for the development of the notochord and the tail in every vertebrate that has one. The gene itself is conserved across vertebrates. The coding regions that produce the working protein are nearly identical in mice, in monkeys, and in humans. What was different was not the gene. It was a sequence of DNA sitting inside one of the introns of the gene, the stretches between coding regions that, until the last 2 decades or so, had been treated as largely inert. In the introns of the ape version of TBXT, Scheer's team found a small mobile genetic element called an ALU-Y. ALU elements are a class of short sequences. The average one is around 300 nucleotides long, that have been copying themselves around the primate genome for tens of millions of years. Most ALU insertions do nothing observable. They land in a region where their presence has no functional consequence, and they persist as passive passengers in the genome. What the ALU-Y in the TBXT intron does is different. It sits opposite an older ALU element called ALU-SX1 in an adjacent intron, and the two of them, oriented in opposite directions, form a structure that pairs across the intervening DNA when the gene is being transcribed. That pairing disrupts the normal splicing process. The mature messenger RNA that comes out of the gene no longer includes one of its coding segments, called exon 6, because the spliceosome, the molecular machinery that assembles the final transcript, cannot reach it cleanly through the paired ALU structure. The resulting protein is shorter than the normal one. It still performs the transcription factor function of TBXT, but with reduced efficiency in certain developmental contexts, particularly in the tissues that form the posterior end of the body. When Scheer's team engineered mice to carry the same intronic configuration that apes have, inserting a comparable ALU element into the equivalent intron of the mouse TBXT gene, a range of tail phenotypes appeared. Some mice developed normal tails. Some developed shortened tails. Some developed no tails at all. The mechanism was confirmed. The ALU-Y insertion, sometime in the common ancestor of apes around 25 million years ago, disrupted the splicing of TBXT in a way that reduced the activity of the protein in tail development, and the individuals that inherited the insertion produced embryos whose tails did not complete their formation. The insertion was an accident. ALU elements copy themselves through a process that has no concern for the functional consequences of where they land, and this particular landing was no different. It was one event, in one ancestor, in one generation, somewhere around 25 million years ago on a continent we cannot now identify with certainty. Whether that ancestor had offspring, whether those offspring carried the insertion, whether the insertion spread through the breeding population over the next several thousand generations, those are questions that depend on variables we cannot reconstruct. What can be said is that the insertion did spread, that the population carrying it eventually gave rise to the lineage that includes every living ape and every living human, and that the tail has not formed completely in any member of that lineage since. The accident was not clean. The same alteration to TBXT splicing that reduced tail development also reduced by a small but measurable amount the reliability with which the posterior end of the neural tube closes during embryonic development. The neural tube is the structure that becomes the brain and spinal cord, and its proper closure in the first weeks of gestation is one of the more delicate operations an embryo performs. In lineages with an intact TBXT, the closure happens cleanly in nearly every developing embryo. In the ape lineage, the same change that took the tail away introduced a small but persistent rate of failure at the posterior closure, the conditions known in their human presentations as spina bifida and sacral agenesis. These are rare. They are not rare enough. Every year, tens of thousands of infants are born with some form of neural tube defect, and the underlying architectural vulnerability, the fragility that makes those defects possible, is the same molecular change that relieved our ancestors of their tails. This is the price. It is not a large price, averaged across the population and the generations. The tail loss presumably conferred some advantage. The leading hypothesis involves the balance requirements of arboreal locomotion in early apes, though the evidence for any specific adaptive story remains contested, and the advantage, whatever its exact shape, was sufficient to maintain the ALU-Y insertion in the lineage across 25 million years of subsequent evolution. The cost was the small chance in each embryo that the posterior end of the developing spine would fail to close properly. Both the benefit and the cost were delivered by the same molecular event, and neither can be separated from the other without undoing the whole arrangement. You carry that arrangement in your body. The coccyx you are sitting on right now is the remnant of a tail that began to form in your own embryo and was taken apart over the course of several weeks in utero because the transcription factor that would have completed it was running at reduced efficiency. The ALU-Y that reduced that efficiency sits, as I am saying this, in one of the introns of one of your TBXT genes on chromosome 6 in every cell of your body that contains a nucleus. It was not placed there for any reason. It was not placed there by any agent. It arrived by the ordinary undirected mechanics of genomic self-copying in the germ cell of an ancestor you and I share with every gibbon and every orangutan and every chimpanzee alive today, and it has been copied into every cell of every descendant of that ancestor for 25 million years. The tail you did not develop, the spine that closed well enough, the small but non-zero risk that it might not have, the coccyx that is currently bearing your weight. These are not separate facts. They are the same fact, viewed from different angles, all of them downstream of a single accident with 300 nucleotides and an unfortunate landing. There is a way to sit with this that is worth taking a moment for. The body we are told about, in most of the accounts that shape how we think about ourselves, is the product of a long process of refinement. Things that work are kept, things that don't work are discarded, and what remains at any given generation is the best adapted version of the organism for the environment it finds itself in. That account is not exactly wrong, but it is not the full picture of what the evidence actually shows. The evidence shows an organism that is an archive of events, some of them selected for, some of them selected against, some of them neither. And the distinction between a feature that was selected and a feature that simply arrived and stayed is not always possible to draw. The loss of the tail was not, in any meaningful sense, a refinement. It was an accident that happened to be tolerated. The spina bifida risk that came with it is not a flaw that selection failed to fix. It is the price of the tolerance, carried forward across millions of generations because the price was small enough, and the alternative was a lineage that did not exist. The coccyx at the base of your spine is 3 to 5 fused vertebrae that were, in your embryo, the beginning of a tail. The tail did not form because an element 300 nucleotides long sits in one of the introns of one of your genes, where it has sat in all of your ancestors going back to a single hominoid ancestor in the late Oligocene. The pelvic floor muscles that hold your organs against gravity are attached to that remnant, a use for an old structure that the body happened to already have, pressed into service because the structure was already there and the muscles needed somewhere to anchor. The organ the tail was does not exist anymore in your lineage. The bone it became is still doing work. The gene that builds it has not been repaired. The accident that broke it has not been corrected. None of this happened because anyone decided it should. All of it is happening right now, inside you, because it has been happening, uncorrected, for 25 million years. The archive of the body is not a record of a plan. It is a record of what was tolerated. What the listener carries in flesh and in nucleic acid is not a designed form. It is the persistent shape of everything that the long blind process did not undo. Some of what the long process did not undo is visible from the outside and has been part of what the body has been quietly doing throughout this documentary. Some of it is further inside, in regions the body conceals from ordinary view. One of the more ordinary of them, ordinary in the sense that almost every human being has encountered its small failures, runs along the back of the throat and into the bones of the face. Consider for a moment the ordinary geometry of a goat. A goat spends most of its waking life with its head oriented roughly parallel to the ground or angled somewhat downward toward the grass it is grazing on. Its spine is horizontal. Its face, when it is feeding, points forward and slightly down. This is the arrangement the goat's ancestors have held for the whole of the evolutionary history that shaped the goat's skull, and it is the arrangement the skull was built for. Inside that skull, in the bones of the upper jaw just below the eye sockets, sit a pair of air-filled cavities called the maxillary sinuses. Each one is roughly the size and shape of a walnut shell, hollowed out and turned sideways, lined with a thin sheet of mucus-producing tissue and connected to the nasal cavity by a single small opening. The opening has a name, ostium, and in the goat, the ostium sits near the lower edge of the sinus, where any mucus or fluid that accumulates inside the cavity can drain out through it by the ordinary action of gravity. The sinus fills slightly, the mucus flows toward the opening at the bottom, and the system clears itself without the animal having to do any particular work to move the fluid along. You carry the same pair of sinuses. Their walls are built from the same bones, lined with the same kind of tissue, connected to the nasal cavity by an ostium of the same basic design. The cavity is somewhat smaller in your case, the contours are a little different, but the structure is recognizably the same structure, inherited from the same mammalian ancestors whose skulls also shaped the goats. There is one difference worth noticing, and it is a difference in the position of the ostium. In your maxillary sinus, the ostium does not sit at the bottom, it sits near the top. The arrangement made sense once. In a quadrupedal mammal holding its head roughly horizontal, an ostium placed on the upper wall of the sinus cavity was pointing, in terms of the animal's actual orientation in space, toward the direction gravity would move mucus. The opening was at the drainage end. The system worked passively. Small volumes of mucus accumulated inside the sinus flowed toward the opening that happened to be in the gravitational low point of the cavity, given the head's habitual posture, and drained out into the nasal passage. This was the system our ancestors carried for the whole of their mammalian history. Somewhere in the last several million years, our lineage stood up. The transition to habitual bipedalism did a number of things to the body's geometry that are, by now, reasonably well catalogued. The pelvis rotated. The spine curved into its characteristic double S. The legs lengthened relative to the arms. The foot became an arched propulsive platform rather than a grasping organ. The head, sitting now at the top of a vertical column rather than the front of a horizontal one, tilted into a new default orientation in which the face points forward rather than forward and down. Many structures adapted to this change in posture, some of them imperfectly. The lower back carrying loads it was not originally shaped to carry, became a chronic source of trouble. The knees, bearing the full weight of the body on two joints that had been designed for four, developed their own set of characteristic failures. The feet flattened or arched, or fell somewhere in between depending on the particular family lineage, and the resulting problems filled the caseloads of podiatrists across the developed world. Inside the skull, the sinuses did not adapt at all. The ostium of the maxillary sinus is still where it was in the quadrupedal ancestor. It still sits on the upper portion of the medial wall, roughly where you would expect a drainage opening to be if the head were held parallel to the ground. In a bipedal human, with the head held vertically above a vertical spine, that position is no longer at the gravitational low point of the sinus. It is near the top. Any mucus that accumulates inside the cavity, instead of flowing downward through the opening by passive drainage, has to be moved upward against gravity in order to reach the exit. The body has a mechanism for this. The lining of the sinus cavity is covered with millions of small, hair-like projections called cilia, and the cilia beat in a coordinated pattern that drives a thin film of mucus across the surface of the tissue toward the ostium. The motion is continuous and automatic, and under ordinary conditions, in a healthy person, it keeps the sinus clear without any conscious effort. The mucus reaches the ostium, passes through into the nasal cavity, and is swallowed or cleared by the ordinary motion of the nose. In a bipedal human, the ciliary system has to do its work against gravity rather than with it, and the margin by which it succeeds is narrower than it would be in the ancestral arrangement. When the system is stressed, by inflammation, by infection, by an allergic reaction that thickens the mucus, by a viral illness that increases the rate of mucus production, the ciliary motion can fall behind and mucus accumulates in the sinus rather than draining out of it. The pressure builds, the tissue swells. The ostium, already a narrow passage, narrows further. The clinical condition that results is called sinusitis, and it is one of the most common reasons adults in industrialized countries visit primary care physicians. How much of the prevalence of sinusitis can actually be attributed to the ostium's position is an argument that still has some air in it. There are two reasonable views, and the honest account involves holding both of them. The first view, which has been the standard vestigial structure reading of the sinus for several decades, treats the arrangement as a textbook example of suboptimal design, a piece of quadrupedal anatomy inherited into a bipedal body, no longer functioning under the conditions it was shaped for and producing predictable problems as a result. The cadaveric studies that have actually measured drainage rates from human maxillary sinuses positioned in different head orientations do support this view to a degree. When a cadaveric human skull is tilted forward into something resembling a quadrupedal posture, the same sinus that drained poorly in the upright position drains measurably better. The ostium geometry on this reading is straightforwardly a legacy inheritance. The second view is somewhat more cautious. It points out that ciliary transport, not gravity, is the primary mechanism of sinus drainage in all mammals, including the goats and other quadrupeds whose ostia happen to sit in the gravitational low point of their cavities. The ciliary system is quite capable of moving mucus against gravity in a healthy human, and under normal conditions it does so without difficulty. An opening placed at the bottom of the sinus would, on this account, create its own problems: too rapid drainage, excessive drying of the mucosa, possibly plugging by debris that would be moved too fast. Many individuals have accessory ostia, additional openings lower down on the sinus wall that provide secondary drainage paths, and the existence of these accessory openings complicates the simple story about a single mispositioned exit. Both accounts have evidence behind them, and both probably contain some portion of the truth. What seems to be the case, when the evidence is weighed honestly, is that the bipedal rearrangement of the skull made the sinus system somewhat more vulnerable to disruption than it had been in our quadrupedal ancestors, without quite making it dysfunctional. The cilia do most of the work. Gravity would do some of it, in a different posture, but the posture is no longer what the anatomy assumes. The result is a system that functions adequately under ordinary conditions, and fails more often than a freshly designed system would under adverse ones. This is not the most dramatic illustration of evolutionary inheritance in the human body. It is, for exactly that reason, one of the more instructive ones. The body does not usually fail catastrophically. It fails when it fails at the margins, in the places where an old arrangement and a new set of demands meet each other at a slight but consistent mismatch. If you have ever had a head cold that would not clear, if you have ever felt the particular pressure behind the cheekbones that signals accumulated fluid with nowhere convenient to go, if you have ever lain down with a stuffed nose and found that changing the angle of your head changed how well the passages drained, you have encountered in ordinary experience the geometry the body inherited from its quadrupedal past and has not quite reorganized since. The arrangement is not broken. It is simply holding the shape it was built to hold, in a body that now holds a different shape itself. Some of the body's older legacies are less consequential than a seasonal sinus infection and involve structures so small that most people go their whole lives without consciously noticing them. At the inner corner of the eye, where the lashes meet the bridge of the nose, sits a small pink fold of tissue whose modern function is minor, a partial contribution to tear drainage, a slight allowance for the eye's rotation, and whose origin lies in an organ that most of the animals we share a lineage with still carry in functional form. The next time you're in front of a mirror, under reasonable light, look at the inner corner of one of your eyes, the corner nearer the nose, where the upper and lower lids meet and where tears collect when they come. Tucked into that meeting point, between the small pink bump called the lacrimal carbuncle and the white of the eye itself, there is a thin, vertical fold of tissue, pinker than the surrounding white, curved slightly like the edge of a half moon. In most lighting, it is easy to overlook. Once you have noticed it, you cannot unnotice it. It is called the plica semilunaris, which is the Latin for half-moon fold, and it has been sitting there doing very little for as long as there have been eyes like yours to carry it. What the plica is a remnant of is not, strictly speaking, another part of the eye. It is another eyelid. In most of the terrestrial vertebrates alive today, in fish, in amphibians, in reptiles, in birds, in a great many mammals, the eye is protected not by 2 eyelids, but by 3. The third lid, called the nictitating membrane, sits at the inner corner of the eye and sweeps horizontally across the surface of the cornea rather than closing down from above or up from below the way the upper and lower lids do. In the animals that still use it, the membrane is usually thin enough to allow at least some vision through it, and it serves 2 overlapping purposes. It protects the eye from physical damage and drying out during moments when keeping the eye fully open matters, diving underwater, striking at prey, flying into the wind, and it clears the surface of the cornea of dust and debris the way a windshield wiper clears a windshield. An owl blinking sideways as it tears into a mouse is using its nictitating membrane. A crocodile sliding under the surface of a river and continuing to watch for movement above the water is using its nictitating membrane. A chicken, drawing a thin veil across its eye between glances at the ground, is using its nictitating membrane. Among the mammals, the membrane is retained in functional form in quite a few lineages. Camels, which spend much of their lives in environments where blowing sand would otherwise scour the cornea, have substantial nictitating membranes that are still active and doing their original work. Polar bears diving for fish through holes in sea ice have them. Manatees, feeding on aquatic vegetation with their eyes open underwater, have them. Cats and dogs still have a reduced but partially functional version of the membrane visible at the inner corner of the eye when the animal is drowsy or unwell. Among the primates, with one extraordinary exception, the membrane is essentially gone. The exception is a small West African primate called the Calabar anguantibo, a nocturnal relative of the lorises, slow-moving, strictly arboreal, about the size of a small cat. The anguantibo retains a fully functional nictitating membrane, one of the only primates alive to do so, and uses it in the ordinary manner of the vertebrates that have not lost the structure. Whether the anguantibo represents the retention of a function the rest of the primate lineage gave up, or whether it represents an unusual case of redevelopment from an already reduced membrane, is not entirely settled. What is clear is that the anguantibo is the exception that demonstrates how comprehensively the rest of the primate line has moved on from the structure. Why the primates let it go is less clear than the fact that we did. One plausible account involves the evolution of the primate eye itself. Forward-facing, recessed into bony sockets that provide substantial mechanical protection, lubricated by a tear film maintained by glands and ducts that can refresh the corneal surface continuously rather than by sweeping. The protective and cleaning functions that the nictitating membrane performs in a chicken or a crocodile are, in a primate, being handled by other means. The old structure was redundant, the selective pressure that had maintained it relaxed, and over the course of tens of millions of years the membrane shrank to the small fold you can see at the corner of your own eye. The reduction was not, it turns out, complete. The plica semilunaris is not simply an inert remnant taking up space in the corner of the eye. It contributes, in a small way, to the drainage of tears. The medial corner of the eye, where the tear film collects and eventually flows into the tear duct, depends on a particular arrangement of soft tissue that allows the fluid to pool slightly and then move through the small opening into the nasolacrimal system that drains it into the nose. The plica's presence helps shape that pooling and its drainage and allows the eyeball to rotate further in its socket than it would if the conjunctiva, the thin membrane that lines the inner surface of the lids and the front of the eye, were anchored more rigidly at that corner. The old structure has not been put to a dramatic new use. It has been absorbed into the ordinary maintenance of the eye, handling small tasks that the eye was managing already and that the plica happens to assist with. Rarely, very rarely, an individual is born whose nictitating membrane did not fully reduce during embryonic development. The membrane is present at the inner corner of the eye in something approaching its ancestral form, thin, partially translucent, capable of at least partial movement. The cases are uncommon enough to be reportable in medical journals when they appear, and they are almost always noted with some care because what they represent is the developmental program for a structure the lineage has not used in tens of millions of years, briefly surfacing in a single individual. The instructions to build the full membrane are not entirely gone from our DNA. They are quieted, modified, redirected, but the underlying capacity to build the structure is still there, and when the quieting fails, the older form reappears. The embryo does not know that its lineage has moved on. It builds what the genes tell it to build. Usually the genes tell it to build a small pink fold. Sometimes they tell it to build something more. What the plica shows, in the quietest possible way, is that the body does not fully erase what it stops using. It reduces, it redirects, it finds small secondary uses for what remains, and it carries the remainder forward in a form that is not functional in its original sense, but is not quite inert either. Almost every time you blink, the plica sits in the corner of your eye, doing its minor work in the drainage of tears, while the vast evolutionary majority of your vertebrate relatives, every fish in the ocean, every bird in the sky, every reptile in the sun, every small West African primate in a forest most of us will never see, sweeps a third lid across the surface of the eye in a motion you have never performed. The eye is not the only place in the head where a small muscle is still carrying out a gesture its lineage lost long ago. Behind the ears, underneath the skin of the scalp, several small muscles remain attached to the cartilage of the outer ear. They are innervated, they respond to commands from the nervous system, they still attempt to perform the motions they were selected for. Motions you have almost certainly, as an adult human being, never intentionally made. In a soundproof laboratory at Saarland University in southwestern Germany, in a room small enough to sit about 6 people comfortably and lined with the foam wedges that absorb reflected sound, a participant is arranged in a chair that keeps the head from moving. The head is held in place by a padded chin rest of the kind used in ophthalmology clinics for the same reason, to prevent the small, involuntary motions that would otherwise contaminate the measurements. Electrodes are attached to the scalp just above and behind each ear, positioned over 2 of the small muscles that connect the outer ear to the skull. The participant is given an audiobook to listen to, played through a set of speakers arranged at specific angles around the room, and is told to pay close attention to the content, because they will be asked questions about it afterward. A second audio track, a podcast on an unrelated topic, is then played at the same time from a different speaker. The participant must follow the audiobook against the interference of the podcast, and the researchers measure moment by moment what the small muscles behind the ear are doing while the participant listens. What the muscles are doing, it turns out, is trying to move the ear. The motion is not visible. If a second person were standing in the room watching the participant from a few feet away, they would see a person sitting in a chair listening to audio, with no observable change in the position or orientation of either ear. The small shifts the muscles produce are on the order of fractions of a millimeter, buried under the mass of the outer ear and the tissues of the scalp, and they do not translate into any movement that would serve the acoustic function they were once shaped for. What the electrodes record is not the motion itself, but the electrical signal that would, in an animal with fully functional ear-directing musculature, produce the motion. The signal is real, the command is being issued, the muscles are contracting, the ear is not turning. The researchers, a team led by a neuroscientist named Andreas Schröer, published their results in the journal Frontiers in Neuroscience at the beginning of 2025. The finding they were most interested in was specific. When the audiobook came from a speaker positioned behind the participant, requiring additional effort to follow against the distracting podcast, the electrical activity in the muscle just above the ear, a muscle called the superior auricular, increased measurably. The harder the participant worked to extract the signal from the noise, and the more the signal was coming from a direction that could not be resolved by simple head orientation, the more strongly the muscle fired. The muscle was not producing useful motion. It was, in the neurological sense, doing the same thing it has always done, responding to the brain's attempt to orient the external ear toward a sound worth paying attention to. The response was complete. The motor output, when it arrived, had nothing to move. The system Schröer's team was measuring is old. Most mammals have it in functional form. Cats rotating their ears toward the scrape of a claw at the back of the house, dogs turning a single ear to catch a whistle, horses and deer and rabbits performing the characteristic swivel that places one or both ears in the optimal position to receive a particular sound. The outer ear in these animals is mobile, often highly so, and the small muscles that move it, the auricular muscles, of which there are 6 principal ones in the standard mammalian layout, are under continuous fine control. An attentive listener can watch a cat triangulate a sound in real time by the independent motion of the 2 ears, and the motion is an extension of the animal's attention rather than a separate system from it. Where the ears point is where the animal is listening. Somewhere around 25 million years ago, in the lineage that would become the hominoids, the system stopped being used for its original purpose. The precise drivers of the loss are not fully understood. The arrival of more flexible neck rotation may have been part of it. Once the whole head could be turned quickly and accurately toward a sound source, the separate orientation of the external ears became redundant. The increasing primacy of visual attention in our ancestors may have been part of it. The reduction of the outer ear itself from the large mobile flap of a typical mammal to the smaller, less mobile structure that characterizes the apes and humans contributed its own feedback to the system. Whatever the sequence, the result is that our ancestors, across the last 25 million years, became animals whose ears do not point. The outer ear became a fixed funnel. The neural machinery that had directed the ear's motion did not, however, disappear. The muscles are still there. The nerves that innervate them are still there. The central commands that were once issued to direct the ear are still being issued routinely, dozens or hundreds of times a day, in every listener who is paying attention to a sound in their environment. What has changed is only the final stage. The muscles receive the signal, they contract, and they produce a displacement too small and too damped by the surrounding tissue to accomplish anything useful. The system is intact except at its end point. Every component works except the one that would make the whole thing matter. A small minority of modern humans, somewhere between 10 and 20%, depending on the estimate and the population, can produce a visible ear motion through voluntary effort. The ability is distributed in families and appears to have some heritable component, though the specific genetics have not been fully worked out. What the ear wigglers are doing neurologically is tapping into the same pathway that fires involuntarily in the Schroer Laboratories participants, but through conscious rather than reflexive command. They are not operating an unusual piece of anatomy, they are operating the ordinary anatomy at an unusual volume, producing enough motor output to overcome the damping and generate a visible result. What the rest of us lack is not the machinery, it is the access. There is a small piece of ear anatomy connected to this story, but with its own history, that is worth knowing about. If you run a finger along the upper curve of your outer ear, along the rim called the helix, you will find, in somewhere between 10 and 60% of people depending on the population, a small thickening of the cartilage. It is not a nodule or a lump. It is a slight protrusion, sometimes visible, sometimes only palpable, located on the inner surface of the helix, about where the rim begins to turn from its upward arc toward the earlobe. The structure has a name, and it has a minor history. It is called Darwin's tubercle, because Darwin wrote about it in The Descent of Man in 1871, though he himself credited the observation to an English sculptor named Thomas Woolner, who had noticed it while working on a bust of the human head, and had recognized it as the vestigial remnant of the pointed ear that mammals such as cats and rabbits still carry. The tubercle is what is left of the fold of cartilage that, in a pointed mammalian ear, runs to the ear's apex. In our lineage, the point receded. The cartilage involved in forming it reduced, and the Darwin's tubercle is the small residual thickening where the point used to be. The tubercle is unusual among vestigial features in that it is polymorphic, present in some people, absent in others, highly variable in size and prominence across populations, and its genetic basis has turned out to be one locus among several associated with ear morphology. A genome-wide association study published a few years ago identified a particular gene involved in cartilage formation, called HAS2, as one of the variants affecting the tubercle's development. The gene is not specifically a tubercle gene, it shapes many aspects of facial and cranial cartilage. The tubercle is a secondary consequence of variation in a gene that does other, more central work in the developing embryo, and whether any given individual has one visible is partly a function of which particular version of that gene they happen to inherit. The point of mentioning Darwin's tubercle here is not that it is independently important. It is that it sits on an ear whose muscles are still, in the ordinary course of your listening to this sentence, receiving commands to move. The whole structure, the pointed mammalian ear, the muscles that rotated it, the tubercle at its tip, the neural pathway that directed its orientation, was a single integrated system once, and our lineage dismantled only some of it. The external shape reduced, the muscles stayed, the directional command pathway stayed. The neural apparatus that would use the information, if the ear could still be oriented, stayed. What the body assembled out of these components over the 25 million years since the system stopped working as designed is a ghost. The machinery is present. The output is not. You are operating it this moment, as you follow the words of this sentence with whatever portion of your attention is still attending to the sound of them, and the small muscles above and behind your ears are firing in quiet, coordinated patterns that go nowhere. What the body retains from its past is usually, like the ear muscles, a structure that has lost its function but kept its form. There is another kind of retention, less about a structure than about a developmental default in which features that were not selected for at all, features that are simply byproducts of how embryos are assembled, persist in every member of the species whether or not the feature is of any use to that particular member. One such feature is shared by every human being, regardless of sex, regardless of whether they will ever produce offspring, regardless of whether the feature has any measurable function in the life of the individual carrying it. It sits on the front of the chest, and everyone has a pair. There is a reflex in how we talk about the body that is worth examining for a moment. When we encounter an anatomical feature and ask what it is for, we tend to assume that there is an answer, that the feature exists because it serves a function, that the function is the reason the feature was selected and the reason the feature persists, and that if we look carefully enough, we will find the function hiding somewhere in the tissue. The question is so natural that it almost disappears into the grammar we use for biology. What are the kidneys for? What is the liver for? What are the hairs in the nose for? The structure is introduced and the function follows, and the relationship between them seems so obvious that it does not need to be examined. The structures the body carries for no functional reason at all, carries simply because, at some point in embryonic development, the instructions to build them arrived before the instructions not to build them, and because once they were built there was no mechanism to unbuild them, are harder to talk about in this grammar. The grammar wants to finish the sentence. The features do not always cooperate. Every adult male carries, on the front of the chest, a pair of nipples. They have the same shape as the nipples on the front of an adult female's chest. They have the same innervation. They have the same sensitivity. They are surrounded by the same pigmented ring of skin, called the areola. They sit at roughly the same positions relative to the sternum. Most of the time, through the whole of an adult male's life, they do nothing. They are not involved in reproduction. They are not involved in nourishing offspring. They are not involved in attracting a mate, at least not in any way that a careful survey of the literature could confirm as selectively significant. Under certain pathological conditions, hyperprolactinemia from a pituitary tumor, certain medications that disrupt hormonal balance, extreme states of starvation followed by refeeding, the tissue underneath them can activate and produce a small amount of fluid that resembles milk. These cases are rare enough to make medical journals when they happen. For the vast majority of male humans across history, the nipples sit on the chest and are inert. The question of what they are for has an answer, but the answer is not of the form the grammar expects. The developmental program that builds a human body is a sequence of instructions executed in a specific order, and the order is not negotiable. In the first weeks after conception, the embryo proceeds through a series of stages that are essentially identical across all mammals, and within those stages, the earliest instructions lay down the most foundational structures: the basic body plan, the head-tail axis, the rough layout of the major organ systems. Sex is not among the first things the embryo decides. For the first 5 or 6 weeks of human development, there is no differentiation along the male-female axis that is visible in the anatomy of the embryo. The tissues that will become the reproductive organs are present but undifferentiated, and the same developmental program is running in every embryo regardless of its chromosomal sex. The program does what it does. It builds the foundational structures according to the instructions that are active in that week, and those instructions include building, on each side of the midline of the chest, a ridge of specialized tissue called a mammary line. The mammary line runs from the armpit down to the groin. Along its length, at specific positions, small buds of tissue form that will eventually differentiate into functional mammary glands. In most mammals, cats, dogs, pigs, mice, several of these buds along the line become active mammary glands in adulthood, which is why those animals typically carry multiple pairs of functional teats rather than a single pair. In humans and the other great apes, only the pair of buds closest to the chest develops fully, and the others regress and disappear during development. Very occasionally, a human being is born with an extra nipple or two running down the line from the armpit to the groin, and these atavistic structures, called supernumerary nipples, usually small and pigmented and often mistaken for moles, are a visible record of the mammary line the species has otherwise reduced to a single pair. What matters for the present question is that this line forms and the buds at the top of it develop into nipples and areolar tissue before the sex of the embryo has started to differentiate. By the time the signal that directs male sexual development begins to take effect, and the signal is a specific one, a gene called SRY, that sits on the Y chromosome and becomes active sometime around the 6th or 7th week of gestation, the nipples have already formed. The SRY gene, when it activates, triggers the development of testes from the undifferentiated gonadal tissue, and the testes begin to produce the hormones that will direct the rest of male-specific development. Those hormones can influence many things that have not yet been built. They cannot unbuild what has already been built. The nipples are already there. They stay. The female typical developmental program, in the absence of the SRY signal, continues through the mammary development sequence and produces the additional tissue that will, much later, enable lactation in response to the hormonal cascade that accompanies pregnancy. The male typical program diverts the developmental trajectory but does not remove what has already been laid down. The result is 2 bodies, one equipped for lactation, one not, that share the same basic chest anatomy because they share the same developmental program up to the point at which the divergence began, and because everything assembled before that point is common to both. There is nothing in this story that required selection. No pressure acted on male nipples specifically. No advantage was conferred by their presence and no disadvantage by their absence, or at least no advantage or disadvantage substantial enough to generate a selective signal strong enough to modify the timing of the developmental sequence. The nipples are there because the developmental program builds them before the signal to differentiate the sex arrives, and because rearranging the timing of either event, making the sex differentiation earlier or the mammary line formation later, would require modifications to a developmental sequence that is highly conserved across all mammals and tightly interlocked with many other processes. The modifications that would be required are not small ones. The consequences of getting them wrong would be catastrophic. Selection cannot make the required changes without breaking things that are more important than the inconvenience of carrying structures that serve no function, and so the structures are carried by every male mammal alive for as long as mammals have existed. The body is full of features of this kind once you know how to look for them. The tailbone that formed from a tail that would not complete is a feature of this kind. The broken gene for vitamin C synthesis is a feature of this kind. The 8% of the human genome that derives from ancient retroviral insertions, most of it inactive, all of it carried in every cell of every human body, is a feature of this kind. What unites them is not that they serve functions nobody has yet identified. What unites them is that the body does not remove what it costs nothing to keep, and that the threshold for keeping is low enough that a very large number of features end up on the carrying side of it. The body is not efficient. It is persistent. It holds on to what it has built, and what it has built is often more than it uses. This is worth sitting with because it changes the shape of the question we started with. When we ask what something is for, we are assuming that the answer exists. For many features of the body, the answer does exist, and tracing the selective pressures that shaped a structure into the form it now has is one of the genuine pleasures of the comparative biological record. For other features, the answer does not exist because the question was never the right one. The feature was built for no reason and has persisted for no reason, and the developmental sequence produced it as a side effect of producing something else, and the body that carries it the same way every body does, quietly and without comment, and without any particular awareness that the feature is there. Some of what the body carries in this way is visible on the surface. Some of it is buried deeper. A considerable portion of it is not anatomical at all, but molecular, sequences of DNA in the genome that are not expressed, or that are expressed in forms that no longer do what they once did, or that sit in regions of the chromosome that selection no longer acts on with any force. One particular sequence, broken in a specific ancestor sometime around 60 million years ago, is carried in every cell of your body, and the breaking of it has had consequences that extend well beyond the biology of the cell. The consequences reached as far as the decks of 18th century ships, and they killed, by conservative estimate, more than 2 million people before anyone understood what was happening. The scurvy began, as it always began, in the mouth. A sailor would wake one morning to find his gums slightly tender, a faint looseness in a tooth that had seemed fine the day before, a trace of blood on the biscuit of his breakfast. Over the following weeks, the symptoms would progress in a pattern every old sailor on every long voyage ship in Europe could have described from memory. The gums would swell and bleed more freely. Teeth would loosen in their sockets until some fell out. Old wounds, even ones that had healed decades earlier in a different life, would reopen. The scar tissue separating along its original lines as if the healing had never happened. Bruises would appear on the arms and legs without any impact to produce them. The joints would begin to ache, then to throb, then to refuse the bending the sailor's work required. And then, usually somewhere between 2 and 4 months after the first tenderness in the gums, the sailor would die. Between the 16th and 18th centuries, this progression killed more sailors than all other causes of death at sea combined. Conservative estimates place the total above 2 million. Long voyages routinely lost substantial fractions of their crews to the disease before reaching their destinations. Some voyages lost more than half. The condition was known to every naval authority in Europe, treated by every ship's surgeon with remedies that ranged from the ineffective to the actively harmful, and no consensus existed about what caused it or how to prevent it. The fact that fresh produce seemed to help had been noted sporadically for centuries. No one knew why. In May of 1747, aboard HMS Salisbury on blockade duty in the English Channel, a naval surgeon named James Lind set up an experiment that had not, before then, been attempted in the systematic form he gave it. 12 sailors on the ship had scurvy at roughly the same stage of progression. Lind arranged them into 6 pairs, gave each pair the same diet in every respect except for one supplement and tracked the outcome. One pair received cider, one received diluted sulfuric acid, sweetened and flavored, which was a standard treatment in parts of the medical literature of the period. One received vinegar. One received half a pint of seawater each day. One received a paste of garlic, mustard seed, horseradish, and gum myrrh. The last pair received 2 oranges and 1 lemon each day for as long as the supply lasted. The supply lasted 6 days. By the 6th day, the sailor in better initial health of the citrus pair was fit to return to active duty. His partner, weaker at the start, was well enough to nurse the others. None of the remaining pairs showed meaningful improvement. The sailors on seawater, the sailors on vinegar, the sailors on the paste of garlic and mustard, the sailors on the sulfuric acid, all continued to deteriorate at the rates the disease normally permitted. The result was not subtle, and it did not require statistical analysis of any modern kind. Anyone watching the 12 sailors over the course of a week could see which pair was recovering and which were not. Lind published his findings in 1753, and the Royal Navy did not adopt the treatment for another 42 years. The delay is a story in itself, involving the economics of provisioning long voyages, bureaucratic inertia, and the persistence of competing medical theories that placed the cause of scurvy in bad air or spoiled food. It was not until 1795 that the Navy began routinely supplying lemon juice to its ships on extended voyages. The effect on the fleet was immediate. Sailors stopped dying of the disease in the numbers they once had. What Lind had demonstrated, without being able to explain it, was that something specific in citrus fruit prevented and reversed scurvy. The specific something was not identified for another nearly 2 centuries. It was isolated in crystalline form in the early 1930s, named ascorbic acid, from the Latin roots for no and scurvy, and confirmed to be the active agent against the disease. Most animals, it turned out, do not need to obtain ascorbic acid from their food. They make it themselves in a biochemical pathway that runs inside their own cells, converting ordinary glucose through a series of enzymatic steps into the ascorbic acid their tissues require. The pathway is present in fish, amphibians, reptiles, birds, and nearly every mammal, in a form that is recognizably the same across lineages separated by hundreds of millions of years. The final step of the pathway, the step that produces ascorbic acid from its immediate precursor, is catalyzed by an enzyme called L-gulonolactone oxidase. Humans cannot perform the step because humans do not produce the enzyme. We do, however, still carry the gene that codes for it. On the long arm of chromosome 8, in every cell of every human body, there sits a stretch of DNA that is, in nearly every respect, the gene that codes for L-gulonolactone oxidase in other mammals. The exons are recognizable. The sequence aligns with the functional gene in mice, in dogs, in cows, in rats. What the human version carries, that the functional versions do not, is a set of specific mutations that render the whole sequence unable to produce a working protein. Several of the exons have been deleted entirely: exon 2, exon 3, exon 6, 8, and 11. And the exons that remain carry additional mutations, including premature stop codons, that would halt the production of the protein even if the missing segments were somehow restored. The gene is not simply inactive, it has been broken in multiple places by multiple independent mutational events, and the breaks have accumulated over time without being repaired. The approximate date at which the first disabling mutation occurred can be estimated by comparing the human pseudogene to the corresponding sequences in other primates. The inactivating mutations are shared across all haplorhines, the branch of primates that includes humans, apes, Old World monkeys, New World monkeys and tarsiers, which means that the first break predates the split of this branch from its sister group. That split occurred somewhere between 60 and 74 million years ago, during the early stages of the mammalian radiation that followed the extinction of the non-avian dinosaurs. The break happened in a single ancestor in that window, in a lineage that would give rise to every haplorhine primate alive today. Whatever that ancestor was, it lived in an environment where fruit and insects supplied enough ascorbic acid that an individual with a broken version of the gulo gene was at no disadvantage compared to its neighbors. The mutation did not have to be good. It only had to be harmless. It was harmless, and so it spread, and every descendant of that lineage has carried it since. Guinea pigs have done the same thing independently. Somewhere around 20 million years ago, in the lineage that became modern guinea pigs, a different set of mutations rendered their copy of the same gene non-functional. The guinea pig's version is broken at exon 5, rather than at the exons our version is broken at, and the mismatch is how the genetic evidence confirms that the primate and guinea pig losses happened independently rather than being inherited from a common ancestor. A similar independent loss occurred in certain lineages of bats, and teleost fish appear to have lost the pathway entirely at an even earlier point. The pattern across all of these lineages is the same. An animal that lives in an environment where vitamin C is reliably available in the diet can tolerate the breaking of the gene that synthesizes it. Selection stops maintaining the gene. Mutations accumulate. Eventually the gene is a ruin, and the organism is obligately dependent on dietary sources of vitamin C for the rest of the lineage's history. This is the reason the sailors died. Every one of those 2 million men carried in every cell of his body the broken gene that had been broken in the common ancestor of the haplorhine primates roughly 65 million years before any of them was born. On land, with access to ordinary diet, the defect was invisible. The vegetables and fruit and fresh meat of a normal diet supplied enough ascorbic acid to keep the tissues functioning. At sea, eating salt pork and hardtack for months at a stretch, the sailors exhausted their tissue stores within weeks, and the consequences of the defect began to express themselves in the gums, in the joints, in the scars that reopened, in the bruises that arrived without impact. Scurvy is the gradual unraveling of the body's connective tissue, which cannot be maintained without ascorbic acid, which the human body cannot produce, because the gene that would have produced it has been broken since the Paleocene. What Lind had discovered, without knowing it, was a compensation. The oranges and lemons in the sixth pair's diet contained the molecule that those sailors' own cells had once been able to make for themselves, and could no longer make, and had been unable to make for the whole of their species' existence. The citrus was not a cure. It was a replacement for a missing function, delivered from outside the body because the inside of the body, 65 million years ago, had lost the capacity to supply it. The Royal Navy's eventual requirement that lemon juice be carried on all long voyages was, in effect, an acknowledgement of a molecular deficiency written into the human genome, compensated for by a specific class of fruit at a specific rate indefinitely. You are compensating for the same deficiency right now if you have eaten any fresh produce in the last week or so. The gulo gene in your cells is, at this moment, exactly as broken as it was in every sailor James Lind treated on HMS Salisbury in 1747. The difference is that you, almost certainly, have had access to some source of ascorbic acid in the last few days, and your tissues are well stocked. You carry, in every nucleated cell of your body, the fossilized ruin of a gene that your ancestors used for most of mammalian history, and that you do not use, and that would kill you within a few months if the ordinary supply of fruit and vegetables were cut off. One broken gene, inherited across 65 million years, with more than 2 million human deaths in its wake. The archive inside the genome holds more than single broken enzymes. Some regions of the human chromosome carry not one disabled gene, but hundreds of them, all disabled within a narrow window of evolutionary time, all associated with a single sensory function that our ancestors once possessed and that we now do not. What our lineage traded for that loss, over roughly the same stretch of history, is the reason you can distinguish the red of a ripe fruit from the green of the leaves around it, and the reason you will never know, except by inference and by analogy, what the world smelled like to the mammals our ancestors had been before they became primates at all. Somewhere in a forest canopy in what is now East Africa, roughly 25 million years ago, a small tree-dwelling primate is trying to decide whether a particular piece of fruit is worth the effort of reaching for. The decision is not trivial. The fruit is hanging from a branch several body lengths away, across a gap the animal would have to cross by swinging. If the fruit is ripe, the energy gained from eating it will more than compensate for the energy spent retrieving it. If the fruit is unripe, the sugars are locked inside tough rinds the animal's digestive system cannot break down, and the swing will have been a net loss. Getting this calculation right, reliably, across thousands of similar decisions every day is one of the central problems of making a living in the canopy of a tropical forest. Most mammals approach this problem the way their distant ancestors did. They smell the fruit. The volatile compounds a ripening fruit releases are specific and informative, a fingerprint of sugars, esters, and alcohols that shifts detectably as the fruit moves from unripe to overripe. And a nose sensitive enough to read the fingerprint can judge the state of a piece of fruit from a considerable distance. A dog, by a conservative estimate, has approximately 1,000 functional receptors in its olfactory epithelium dedicated to the detection of volatile compounds, and the neural architecture that processes the information from those receptors occupies a substantial fraction of the canine brain. Whether a given dog is interested in fruit or not, the sensory equipment is present and working, and a dog that wanted to assess ripeness at a distance would have the tools to do it. The primate we started with had those tools too once. The olfactory system in early mammals was extensive, sensitive, and occupied a significant portion of the sensory and neural real estate the body devoted to making decisions about the world. For the first 100 million years or so of mammalian evolution, smell was the dominant distance sense. The earliest mammals, small, nocturnal, often living in burrows or in the underbrush of dinosaur-dominated ecosystems, depended on their noses to locate food, to detect predators, to identify mates, and to read the chemical signatures of the territory around them. The architecture of the primate brain inherits that ancestry, and the olfactory bulb still occupies a region at the base of the frontal lobe that hints at what it used to be. What happened somewhere between 30 and 20 million years ago was that a subset of primates in Africa acquired something their ancestors had not had. A duplication event on the X chromosome produced a second copy of a gene for a photoreceptor protein, a gene for what is called an opsin, and the duplicate version, through a series of subsequent mutations, came to respond to a slightly different wavelength of light than the original. The result, in the animals that inherited both copies, was a visual system that could discriminate between red and green in a way the ancestral mammalian eye could not. The old two-color system had been capable of distinguishing light from dark and blue from yellow, but red fruit against green leaves had read, in that earlier system, as a single color with slight differences in brightness. The new three-color system saw the fruit and the leaves as distinctly different. The signal that smell had been providing, slowly and at limited distance, was now available instantly across the visual field. The advantage was substantial enough to be maintained, across the lineage that would become the old world monkeys and apes for the entire subsequent history of those animals. Every catarrhine primate alive today, every baboon, every macaque, every gibbon, every chimpanzee, every human, sees red and green as distinct colors and uses that discrimination, whether consciously or not, in ways ranging from the assessment of food to the reading of facial expressions. The sensory apparatus that had been evolved over 100 million years of mammalian history to solve certain kinds of problems through smell began to be supplemented, and for many of those problems replaced by a visual apparatus that solved them faster and at greater distances. When a sensory system stops being the primary one a lineage relies on, the genes that build the system come under a different kind of selective pressure. The genes are not actively harmful. They continue to function as they always have, producing the receptors and supporting proteins they code for. But the strength of selection maintaining them, the degree to which individuals carrying a slightly degraded version of one of those genes are at a disadvantage compared to individuals carrying the functional version, weakens. Mutations that would have been filtered out in an earlier ancestor are tolerated. Mutations that would have been disastrous remain disastrous, but mutations that disable individual receptors without compromising the overall system accumulate. Across millions of generations, in a lineage where smell has become secondary to vision, the olfactory gene family begins to erode. You can read the erosion directly in the modern human genome. The olfactory receptor gene family in mammals is large, approximately 1,000 genes in most species, each one coding for a receptor tuned to a particular range of volatile chemical signatures. In mice, which rely on smell the way our pre-primate ancestors did, the overwhelming majority of these 1,000 genes are functional. Somewhere around 20% carry mutations that have rendered them non-functional, disabled by the same kinds of small errors that disabled the gulo gene in our haplorhine ancestor, and the rest remain active. In humans, the proportions are roughly reversed. Over 60% of our olfactory receptor genes are disabled. Approximately 600 receptors that our mammalian ancestors used are, in the current human genome, broken in various ways: premature stop codons, shifted reading frames, missing segments, and carried without being expressed or being expressed only in truncated forms that do not reach the cell surface. The 400 or so functional receptors that remain are enough for us to distinguish several thousand odors, which is a great many, but it is an order of magnitude fewer than the mammalian baseline and perhaps 2 orders of magnitude fewer than the repertoire available to a specialist olfactory animal like a dog. This is one of the 2 molecular records of the trade our ancestors made. The other record is anatomical, and it sits inside the front of your skull, in a location most people have never consciously examined. Most mammals have, in addition to the main olfactory system of the nose, a secondary chemosensory structure called the vomeronasal organ. It is a small paired structure, shaped something like a thin tube on each side of the nasal septum, with its own dedicated receptor cells and its own neural pathway to a dedicated region of the brain called the accessory olfactory bulb. The vomeronasal system does not process general odors the way the main olfactory system does. It processes a more specific class of chemical signals, the pheromones used in intraspecific communication, particularly around reproductive and territorial behaviors. When a cat curls its upper lip and holds its mouth slightly open after investigating a scent, the posture is called a flamen response, and what it is doing is directing air across the openings of the vomeronasal organ on the roof of its mouth. Horses do this, deer do this. Most mammals that still use the system display some version of the gesture. In humans and in the other catarrhine primates, the vomeronasal organ has become something between a reduced structure and an absent one. Some anatomists can identify small pit-like depressions on either side of the nasal septum in human adults and argue that these represent a residual vomeronasal organ, though others dispute whether what is being identified is a functional remnant or simply normal anatomical variation in an area that no longer contains the organ at all. What is not in dispute is the genetic record. The gene that codes for a critical ion channel called TRPC2, which is required for the vomeronasal system to function in every mammal that still uses one, has been disabled in humans and in every Old World monkey and ape. The disabling mutations are shared across the whole catarrhine lineage, which means that the inactivation occurred in the common ancestor of that lineage and was carried forward ever since. The window in which the inactivation occurred corresponds, to a degree that is unlikely to be coincidence, to the window in which the red-green color vision trait became established. The two events happened, as far as the molecular dating can resolve, at roughly the same time in the same ancestor. You cannot perform the phlegmon response, not because you lack the facial muscles, but because the organ the response would direct air toward has, in your ancestry, been gone for 25 million years. The neural pathway that would process the signals it collected no longer exists in functional form. The gene that would run the receptors has been broken for approximately as long. The chemosensory world the vomeronasal system detects, the world of pheromonal communication that other mammals routinely navigate, is, for you and every other catarrhine primate, entirely inaccessible. When another human being enters a room, you can see them, you can hear them, you can perceive whatever odors they carry strongly enough to register on your reduced olfactory system, and that is all. Whatever chemical information they might be producing at the level most other mammals routinely read, you do not have the equipment to receive. What your lineage acquired in exchange, the capacity to see a red fruit against green leaves, the capacity to read the small color shifts in a human face that signal blush or pallor, or the beginning of an emotional response, the capacity to distinguish hundreds of millions of fine color gradients that earlier mammals would have registered as slight variations on a single hue, is what you are doing right now if your eyes are open to any colored surface in the room around you. The trade was not, as far as any evidence suggests, consciously made by any ancestor. No primate walked away from the old sensory system. The new visual capacity arrived by duplication and mutation, and in the lineage where it arrived, the individuals that possessed it reproduced at slightly higher rates than the individuals that did not, and the balance of sensory attention within the whole species' brain began to tilt. Over millions of years, the olfactory genes accumulated the small deleterious mutations that an earlier selection would have filtered out, and the vomeronasal system quietly eroded into the non-functional remnant we now carry. The lineage did not lose smell suddenly. It lost smell gradually because it no longer needed smell as much. The trade is not, in any strict accounting, complete. We still smell things. The residual 400 functional olfactory receptors provide a perceptual world rich enough for perfume and wine and fresh bread to register as complex and pleasurable sensations, rich enough for burning plastic to trigger an immediate alarm response, rich enough for the olfactory industries to constitute a major part of the modern economy. What we do not have is the earlier, more detailed, more urgent world of smell that our mammalian ancestors lived in and that our canine companions still inhabit. We traded most of it. We did not intend to trade it. The trade happened because selection stopped maintaining a set of genes whose products had become less central to survival than they had been for the previous 100 million years, and the genes, unmaintained, drifted. Some of what the body carries from its evolutionary past was never a whole organ or a whole sensory system. Some of it is smaller, individual stretches of DNA that entered the genome from outside the lineage entirely millions of years ago and have been carried inside it ever since. a substantial portion of what you are about to carry forward through the remainder of your life is, in a specific and precise sense, not originally ours at all. In the year 2000, a team working in Somerset, Massachusetts at a biotechnology company called Genetics Institute published a paper in the journal Nature describing the function of a gene they had been studying for several years. The gene coded for a protein that was expressed almost exclusively in the human placenta, and specifically in a layer of that organ called the syncytiotrophoblast. The syncytiotrophoblast is one of the more unusual tissues in the human body. It is composed not of separate cells, each with its own membrane and nucleus, but of a single continuous sheet of fused cytoplasm containing many nuclei spread across the interface between the maternal blood supply and the developing fetus. The sheet is what the mother's blood actually touches when it delivers oxygen and nutrients to the placenta, and what the fetus depends on for every molecule of glucose and every atom of iron it acquires during the 9 months of gestation. Without the sheet, the fetus cannot receive what it needs from the mother. Without cell fusion producing the sheet, there is no sheet. And the protein the Genetics Institute team had been studying was the one that performed the fusion. They called the protein syncytin after the structure it helped produce, and when they examined its amino acid sequence and compared it against the database of known protein sequences, they found the match almost immediately. Syncytin was not a novel human protein evolved for placental function. Syncytin was, in every respect that could be measured, its structure, its mechanism of action, its amino acid sequence, the chromosomal organization of the gene encoding it, a retroviral envelope protein. The specific kind of protein that a virus uses to fuse its own envelope with the membrane of a host cell in order to deliver its genetic material into that cell and infect it. The protein that every placental mammal on Earth uses to form the interface between mother and fetus is, in its molecular biography, the same kind of protein that HIV uses to invade a T cell. The implications of this took some time to settle. What the team had identified was not a gene that resembled a viral gene. It was a viral gene sitting in the human genome, fully integrated into the cellular machinery, expressed at the right times and in the right tissues to build the placental interface, and not doing any of the things a viral gene ordinarily does. It was not being packaged into viral particles. It was not replicating. It was not moving horizontally between individuals. It was acting as a normal human gene, inherited vertically from parent to child, and had been acting that way for as long as anyone could trace. The gene sat in a specific region of chromosome 7, had been there for millions of years, and was doing load-bearing work in one of the most fundamental processes of mammalian reproduction. Whatever it had once been, it was now a human gene, and whatever virus had contributed it to the human lineage had done so a very long time ago. The background to this finding, though it was not widely known outside of specialist circles, had been developing for most of the previous decade. Beginning in the early 1990s, genomic sequencing had made it possible to survey the total DNA content of an organism's cells and catalogue what was there. What the surveys revealed was unexpected in its magnitude. A substantial fraction of the human genome, something in the neighborhood of 8%, consisted of sequences that appeared to be of retroviral origin. The sequences had the characteristic structure of retroviral elements. 2 long terminal repeats flanking a central region that contained, in various states of degradation, the core genes a retrovirus uses to reproduce itself. Most of these sequences were clearly non-functional. The coding regions had accumulated stop codons and deletions across millions of years and could no longer produce working proteins. But the sequences were there, carried in every cell of every human body, inherited through the germline from ancestors who had been infected and whose offspring had inherited the integrated viral DNA as a permanent addition to their own. The process by which this happens is not complicated, though it requires a particular set of circumstances. A retrovirus, when it infects a cell, reverse transcribes its RNA genome into DNA and integrates that DNA into one of the host cell's chromosomes. For almost all infections, the affected cells are somatic, ordinary body cells, and when those cells die, the integrated viral DNA dies with them. Very occasionally, though, the infected cell is a germ cell, a sperm or egg cell, or one of their precursors. And when that happens, the integrated virus becomes part of the germline. The offspring that results from fertilization involving that germ cell carries the viral DNA in every cell of its body, and so do its descendants, indefinitely. The virus has, in effect, moved from being an external pathogen to being an internal component of the host lineage. It is no longer infecting, it is being inherited. If its presence is not strongly deleterious, it persists. If its presence confers any advantage, however small, it is favored by selection. And if the viral coding regions accumulate enough mutations across enough generations to render the original viral functions inoperative, the element becomes what geneticists call an endogenous retrovirus, a fossilized fragment of an ancient infection, carried forward in the genome long after the active virus that produced it has gone extinct. 8% of your genome is material of this kind. To put that figure in perspective, the total fraction of your genome that codes for protein, the genes that actually produce the enzymes and structural proteins your cells depend on to function, is approximately 1.5%. The endogenous retroviral content of your genome is, by mass, more than 5 times the size of your protein coding content. For every gene you inherited from your mammalian ancestors, you inherited roughly 5 sequences worth of viral DNA from infections they and their ancestors sustained at various points across the last several hundred million years of animal evolution. Most of this material does nothing. It sits in the genome, largely silent, accumulating mutations at the neutral rate that uncoded DNA accumulates mutations, providing a kind of dating record that researchers can use to reconstruct when particular infections entered the lineage. A smaller portion of the viral material has been co-opted into various regulatory functions. The long terminal repeats, in particular, can act as promoters or enhancers for nearby genes, and a number of them have been pressed into service in this role. A much smaller portion still has been co-opted for structural or functional purposes in the organism's biology, and syncytin is the most dramatic example of this, but it is not the only one. There is a second syncytin in humans called syncytin 2, derived from a different ancient viral infection and involved in different aspects of placental function, which was identified a few years after the first. There are syncretins in mice, which turn out to derive from yet another viral source and are specific to the rodent lineage. There are syncretins in carnivores, in ruminants, in the primitive mammalian order Afrotheria that includes elephants and tenrecs. Each of these syncytins performs the same basic function, the fusion of cells at the maternal-fetal interface that produces the placental interface, and each of them was, in its molecular origin, a retroviral envelope protein captured from an infection that occurred in the ancestors of that lineage. The captures were independent. Different viruses, different ancestors, different points in mammalian history, all converging on the same solution to the same problem, because a protein that evolved to fuse a virus to a cell turned out to be useful, in exactly the same mechanical sense, for fusing one cell to another. This is one of the more extraordinary findings in the molecular biology of the last few decades, and it has a consequence that is worth stating directly. Placental mammals, the entire group that includes humans and every animal most of us have ever seen or petted, and the overwhelming majority of the mammalian species alive today, depend on viral proteins for the central act of their reproductive biology. The placenta, which is the organ that makes mammalian live birth possible, is assembled using molecular machinery that our ancestors did not invent. They acquired it from infections. And the infections, integrated into the germline, became part of what it means to be a placental mammal. During the months you were gestating inside another human being, connected to that human being through a tissue whose structural integrity depended on a protein whose ancestors were viral. The interface across which oxygen crossed from her blood to yours, glucose crossed from her blood to yours, every molecule of iron and every nutrient and every antibody crossed from her blood to yours, was formed by a fusion protein derived from a retrovirus that infected a mammalian ancestor tens of millions of years before you or she were born. The virus, whatever it was, is extinct. What remains of it is integrated into your chromosome 7 and into the chromosome 7 of every human being you have ever met, and it expressed itself in the tissues of your own placenta to assemble the interface that kept you alive through 9 months of gestation. If you have children, the same gene will express itself in their placentas. If they have children, in theirs. The inheritance is not of a property we developed. It is of a property an ancient infection developed, which our ancestors acquired, and which we have been carrying, expressed and functional and essential ever since. The category of evolutionary leftover this belongs to is different from anything we have looked at so far. It is not a structure that was selected for and then lost its selective context. It is not a gene that broke and was no longer maintained. It is something that was never ours to begin with, that entered the lineage from outside it, and that has been naturalized so thoroughly that the body that carries it has no way of recognizing that any part of itself was, once, not part of itself. What the body is, at the level of its genome, is not a single unified lineage of inheritance, but a composite, a record of everything that has managed to get into the germline and stay there, whether by being made from within or by arriving from without. The archive is larger than the species that carries it. The archive is also larger than this body. What we have been examining so far has, with only a few exceptions, been contained within the human organism. The structures and genes and sequences that the listener is carrying in their own flesh right now. The principle, though, extends outward. Every lineage of vertebrate life on Earth carries some version of this kind of record, and some of the clearest examples of it are not hidden inside bodies at all. Some of them lie in the ground, in specific places where the transitional forms of ancient lineages can still be seen. About 150 kilometers southwest of Cairo, on the western edge of the Fayum Depression, there is a stretch of desert that was, in the late Eocene, the floor of a shallow tropical sea. The sea is called, in the geological literature, the Tethys, and for most of the history of life it sat between the proto-continents that would eventually become Africa and Eurasia, warm and shallow, full of the kinds of fish and invertebrates that warm, shallow seas tend to produce. Over the last 40 million years, the Tethys has drained and been replaced by the Mediterranean to the north and by great stretches of desert to the south. The Egyptian portion of what was once seafloor is now among the most arid landscapes on Earth. It is called Wadi Al-Hitan, which is Arabic for the Valley of the Whales, and what lies exposed on its surface, visible from the trails that wind through the sandstone, is what the last several million years of Eocene life deposited on the floor of the sea before the sea went away. What it deposited in considerable numbers was whales. The first fossil was described in 1879 by a German botanist named Georg August Schweinfurth, who was exploring the Egyptian interior and came across large vertebrae weathering out of the rock. The identification of what the vertebrae belonged to took some time to settle, and the animal eventually named from the fossils was misidentified in a way that has stuck to it ever since. A paleontologist named Richard Harlan, working from initial specimens in the 1830s, interpreted the bones as belonging to some kind of gigantic marine reptile and named the animal Basilosaurus, king lizard. The name was wrong. Basilosaurus was not a reptile. It was a whale, one of the earliest for which nearly complete skeletons have ever been recovered, and by the time later researchers sorted out its true identity, the name had been in the taxonomic record long enough that changing it would have been more trouble than it was worth. The king lizard, to this day, is a whale. What Basilosaurus was doing in the Tethys 40 million years ago was hunting. The animal was long and serpentine, up to 21 meters from snout to tail fluke, with jaws full of teeth shaped for shearing flesh from larger prey. It was the apex predator of its environment. It was also, at that moment in the evolutionary history of cetaceans, a transitional form. Not quite the streamlined, fully aquatic whale that modern species represent, but well past any recognizable resemblance to the terrestrial ancestors from which its lineage had descended. The body had become fully committed to marine life. The nostrils had migrated partway toward the top of the skull. The forelimbs had shortened into paddle-like flippers. The spine had developed the specialized mobility that cetacean locomotion depends on, driving the body through the water with horizontal flukes at the end of the tail. In almost every respect, Basilosaurus is what a functional whale looks like, stretched out to an extreme length, swimming through a Mediterranean-era sea. In almost every respect. In 1989, a team of paleontologists led by Philip Gingerich of the University of Michigan, working at Wadi Al-Hitan under a permit from the Egyptian Geological Survey, uncovered something that the earlier excavations had not quite found in the same completeness. What emerged from the sandstone, as they brushed the surface clean with the small tools paleontological excavation requires, was a hind limb, a complete one. Articulated to the pelvis of the animal, connected through a hip joint to a femur, the femur joining a knee joint, the knee joining a tibia and fibula, and the tibia articulating with a small structured foot. 4 toes, arranged in the manner of a walking tetrapod. The whole limb was roughly 50 centimeters long, on an animal over 20 meters in total length. The limb was not, by any reasonable biomechanical assessment, capable of supporting the animal's weight. It was not functional for walking, but it was structurally complete. The thigh could flex at the hip, the knee could bend, the toes were articulated. Whatever Basilosaurus was using the limb for, and the current best guess involves mating positioning, the limbs grasping the partner during copulation in the water, it was still there, in recognizable form, on an animal that had otherwise committed fully to life in the sea. Gingerich's find was not unique, as it turned out, but one of the earliest clearly documented examples. Subsequent excavations at Wadi Al-Hitan and at related Eocene sites in Pakistan and North America have uncovered multiple Archaeocete whale skeletons, Basilosaurus and its close relative Dorudon, along with earlier transitional forms like Pachycetus and Ambulocetus, and the overall picture they provide is of a lineage caught in the middle of a transition that took roughly 10 million years to complete. The earliest forms, from around 50 million years ago, are mostly terrestrial mammals with some aquatic adaptation. The later forms are mostly aquatic animals with some residual terrestrial anatomy. The middle forms, of which Basilosaurus is among the most spectacular, are unambiguously marine animals that nonetheless still carry, in reduced form, the hind legs their ancestors walked on. What Basilosaurus did not do was throw the legs away. A creature already committed to aquatic life, 20 meters long, with no observable capacity to use its hind limbs for walking, could reasonably be expected to have simply lost the limbs altogether. Selection, operating on the streamlined body plan that made this animal a successful marine predator, would seem to have no reason to maintain the small, awkward legs still dangling from its pelvis. But the legs were not gone. They had shrunk. They had simplified. They had lost much of what they once did. What they had not done was disappear. And in the subsequent tens of millions of years, as the whale lineage continued its transition into the forms we recognize today, the legs kept shrinking and kept simplifying without ever fully going away. Modern whales, which separated from the Basilosaurus lineage roughly 35 million years ago, still carry the vestigial hind limbs as small paired bones embedded in the body wall, disconnected from the spinal column, no longer articulated to any external structure, visible only on dissection or on careful imaging of the living animal. They are the same bones that Basilosaurus carried in fully articulated form. The femur is still there, the tibia is still there. The foot bones are reduced to small nodes, sometimes unrecognizable, but the developmental program that produced the articulated limb in Basilosaurus produces, in a modern humpback or sperm whale, a reduced version of the same structure. The lineage did not erase what it stopped using. It reduced, it simplified. It tucked the old structure inside the body wall, where it no longer interfered with the streamlined form the animal now depended on, and it carried the bones forward through every subsequent generation for the next 40 million years. The pattern here should be familiar by now. It is the same pattern that runs through the palmaris longus in the human wrist, through the recurrent laryngeal nerve in the human chest, through the coccyx at the base of the human spine, through the plica semilunaris in the corner of the human eye, through the auricular muscles behind the human ear. The principle is the same across every example. Selection can remove a structure only when keeping it costs the organism more than losing it would. Selection cannot remove a structure simply because the structure has outlived the function it was originally selected for. And because most vestigial structures are cheap to carry, a few grams of tissue, a short stretch of DNA, a small region of the developing embryo, they are rarely costly enough to generate the pressure that would remove them. They accumulate, they persist, they drift slowly across millions of years toward reduced forms that continue to be carried because there is no longer any reason either to maintain them or to be rid of them. What Wadi Al-Hitan gives us, that the human body by itself cannot quite give us, is the middle of this process laid out visibly on a landscape. A 20-meter whale with functional-looking hind legs that were not actually used for walking. A transitional form, captured by the ordinary processes of fossilization on a particular stretch of sea floor during a particular few million years of the late Eocene, and now exposed to the air because the seafloor is no longer a seafloor. The archive the human body carries is molecular and anatomical, and most of it is invisible to anyone not specifically trained to find it. The archive Wadi Al-Hitan carries is geological and structural, and it is available to anyone willing to walk across the desert. What it shows is not different in kind from what the body shows. It is the same record, written in a different medium, on a different scale of time. Every lineage of vertebrate life on Earth carries records of this kind. The whales carry their legs. The snakes carry reduced pelvic girdles from the limbed lizards they descended from. Flightless birds carry reduced wing bones from ancestors that could fly. Cave-dwelling fish carry the developmental programs for eyes that no longer form. The body you have been walking around in for the length of this documentary is carrying its own version of the same record at its own scale through its own lineage. And the principle that produced all of these records is the same principle. Nothing erases what it costs nothing to keep. The principle is small when it is stated plainly. Selection cannot design. Selection can only modify what a lineage is already carrying, generation by generation, by keeping the variants that reproduce at slightly higher rates and letting the others fade. What comes out of that process over hundreds of millions of years is not a plan. It is an accumulation. It is what the lineage has built, and what the lineage has stopped building, and what the lineage has tried to unbuild and failed to finish unbuilding, all layered together in a single body at a single moment, with no single architect anywhere in the sequence. The body you are listening with is what that process currently looks like. At the inside of your wrist there is a muscle, or there is not, depending on which lineage of human beings you descended from and which mutations that lineage accumulated on its way to you. The muscle, when present, does almost nothing. It held branches once in the hands of ancestors you will never meet, in forests that no longer exist. Beneath the skin of your forearm, in the 90% or so of people who carry it, it is still present as a thin cord, ready to be detected by a small test with the fingers, ready to be harvested by a surgeon if another muscle elsewhere in the body needs repair. In the 1 in 5 who do not carry it, there is nothing at the wrist to look at, and nothing to test for, and nothing to harvest. Either outcome is the ordinary condition of a structure partway out of the species. At the base of your hair follicles, across nearly every square centimeter of your skin, small strips of smooth muscle are oriented and innervated as they have been in mammals for roughly 200 million years. When the temperature drops, or when something moves through the attention that the body recognizes as significant, those muscles contract. The hair rises. Nothing insulating happens because the fur the response was shaped to lift is not present in the quantities that would make lifting meaningful. But the nerves fire, and in firing, they deliver a signal to the stem cells at the base of the same follicles, and the stem cells respond as they have always responded, by becoming slightly more ready to divide and produce new hair. The system the original selection abandoned is not idle. It has been put to other work by a process no selection specifically arranged. From the base of your brain, a nerve descends into your chest, passes beneath the arch of your aorta, loops back on itself, and climbs up through your neck to reach your voice box. Every word you form requires this signal path. The path is approximately 50 centimeters long. The direct route from brainstem to larynx would be approximately 5. The difference has been accumulating in one form or another for roughly 400 million years, ever since a vertebrate ancestor with gills began the long evolutionary process that eventually produced animals with necks. In a giraffe, the path is 5 meters long. In the longest sauropods that ever lived, it may have been nearly 30. The route you carry is identical in architecture to the route the giraffe carries and the route the sauropod carried, scaled only to the amount of body the route has to cross. At the junction of your small and large intestines, a small blind pouch of tissue hosts a reservoir of the bacterial species you depend on for digestion and immune function. The pouch has been evolved independently in various mammalian lineages at least 32 separate times, which is not the signature of a vestige, but of a solution the mammalian body arrives at repeatedly. What Darwin called a leftover is, on current evidence, a structure being actively maintained by selection for reasons 19th century anatomy could not have identified. In your mouth, or where your wisdom teeth were removed, or where they never formed in the first place, 2 developmental programs have been out of synchronization for about 2 million years. The jaw shrank under the pressure of a new way of processing food. The tooth count has not quite caught up. In most individuals alive today, the result is a structural mismatch that surgery or congenital absence has to resolve. At the base of your spine, 3 to 5 fused vertebrae are all that remain of a tail that began to form in your embryo and was taken apart in the weeks that followed. The tail was stopped 25 million years ago by the insertion of a small mobile genetic element into the intron of a transcription factor. The element is still there, in every cell of your body. The tail does not form. The coccyx carries your weight when you sit. A small elevated risk of a neural tube developmental failure, inherited from the same molecular event, is the continuing price of that long-past accident. In the bones of your face, a drainage arrangement designed for a quadrupedal head sits inside a bipedal skull. It works most of the time. When it fails, the failure is a sinus infection. At the inner corner of your eye, a small pink fold of tissue is what remains of a third eyelid that most vertebrates alive today still use. Behind and above your ears, a set of small muscles fires when you strain to listen, attempting to orient an outer ear that has not moved in response to those signals for 25 million years. On the front of your chest, a pair of nipples sits as a developmental default, built before the signal that would direct the body toward male or female form ever arrived, carried by every human being regardless of whether the feature will ever be of any use. In every cell of your body, on the long arm of chromosome 8, a gene for an enzyme our ancestors once used to make ascorbic acid is broken in multiple places, has been broken for roughly 65 million years, and is the reason you need oranges. In every cell, scattered across your genome, sit 600 broken olfactory receptor genes, the molecular record of a sensory trade your lineage made when the emergence of color vision began to make the old olfactory precision less necessary. In every cell, on chromosome 7 and many other locations, sit the remnants of ancient viral infections that entered your lineage millions of years ago and were naturalized so thoroughly that one of them builds the placenta every placental mammal has depended on since. 8% of the material in your cells is viral in origin. 1.5% is protein coding inheritance from your mammalian ancestors. The genome is, among other things, a much larger archive than the organism that carries it. This is what the process has built so far in you. The accumulation goes deeper than the vertebrate lineage. The cellular machinery that runs everything in your body, the ribosomes that translate your genes into proteins, the mitochondria that produce your cellular energy, the basic biochemistry of metabolism that every cell performs, these are inherited from ancestors much older than any of the anatomical features we have traced. The mitochondria themselves are descended from free-living bacteria captured by a larger cell roughly 2 billion years ago and never digested. The ribosome is older than that, dating to a period before the split between the major domains of life. The genetic code itself is older still. When you go far enough back, the distinction between what is yours and what has been incorporated from outside becomes impossible to maintain. The body is not a single lineage. The body is every lineage whose inheritance has persisted long enough to arrive at this moment in this combination, in this particular instance. There is a way to frame this that is worth taking a moment with, and it is the frame the documentary has been working toward from the beginning. The thread the channel is named for is not a metaphor. It is a physical chain of cellular division running from the first self-replicating molecules on Earth to the cells that make up your body right now, and it has not broken, at any point, across the 3.5 to 4 billion years of life's existence. Every ancestor you have ever had, without exception, lived long enough to reproduce. This is not a claim about their worthiness or their luck. It is the only possible claim because any ancestor who had not reproduced would not be an ancestor. The chain that runs through them runs through cells, generation by generation, without interruption. Not a single generation skipped. Not a single lineage of cells paused. For as long as there has been anything recognizably alive on the surface of this planet, The line that ends at the cells you are listening with has been continuous. You are the current end of it. This is the part of the frame that is worth sitting with, because it does not often get said in the terms the evidence actually supports. The frame does not claim that your life has meaning in any cosmic sense. It does not claim that the process had any destination in mind, or that your existence was intended, or that anything like intent exists anywhere in the system that produced you. The frame claims only that the thread runs. It ran before you. It runs through you. It will run past you if your lineage continues into whatever carries it forward. The archive your body carries is the current state of a record that has been accumulating for longer than the rocks on this planet have held their present positions, and the record continues to be written in every cell that divides inside you, in every gene that is copied from parent to child, in every small mutation that selection will either keep or let fade across the generations to come. Nothing about this is static. Nothing about this is finished. The body you are listening with is the current interim report on a process that has no interim and no report, and the process will continue for as long as the thread continues. The thread has not, at any point in the last several billion years, broken. It is not broken tonight. Outside, wherever you are, the ordinary darkness of a night has arrived or is arriving. Inside the body, the ordinary slow reduction of activity that precedes sleep is doing what it does. The breathing lengthening, the small muscles of the face relaxing, the attention moving inward toward whatever will fill it next. None of the processes this documentary has been describing have stopped. The erector pili in your skin are still ready to fire if the temperature drops. The recurrent laryngeal nerves in your chest are still carrying signals to the larynx. The genes on chromosome 8 are still broken. The syncytins are still expressed in the appropriate tissues, in the appropriate individuals. The coccyx at the base of your spine is still bearing whatever weight you have placed on it tonight. The thread runs through all of this, and through you, and past you, continuously. You can sleep now. The archive continues to be written. The thread continues to run. Rest now.